So we still have 7 minutes 30 seconds to go. A bit of a patience. All the finalists are with us. This is good. Most of the committee is also with us. Perfect. Some of the nominators are with us. That's very good. Everything was easy. And now I even do not steer my own voice. So people tell me you are muted or you are not muted. Or don't talk, for example. Please don't talk. So it's fun. It's the first time, so it's always fun. Which hand you mean? Sorry, I do not touch the keyboard, so I'm just. I didn't know that. One thing is certain, that we cannot speak all together because that would be a big mess. There will be, yeah, we should make something like, I should actually send you some food.
Welcome at Dream Chemistry Award 2020 Finals, the competition organized by Institute of Physical Chemistry of Polish Academy of Sciences and Institute of Organic Chemistry and Biochemistry of Czech Academy of Sciences. First of all, welcome our dreamers, our finalists, Claudia Bonfire, Ivana, Christopher Hendon, Yunyan Q, and Pavel De Dio. They dreamed their dreams, but there is only one prize for them. Why they dream in chemistry? Because the chemistry crosses the borders of disciplines, but after all, the chemistry is the central science. Let's welcome our supporters, the members of honorary committee, Richard Szrok, Krzysztof Matejaszewski, Bartosz Grzybowski, and Józef Michel. Someone has to decide who will win the prize, so let's welcome our 18 referees of this year's competition. Let's welcome our counselor who advised us that this prize is very specific, like a Nobel Prize, and you have to be nominated. Let's welcome our nominators. They include Sir Fraser Stoddart and Mirza Dinka, who is actually the Dream Chemistry Award winner. Finally, let's welcome our special guest, Russell Johnson, Nature Chemistry editor. I'm sure that every chemist dreams about publishing in Nature, so he will tell us how to do it. So let's welcome everybody and good luck for our Dreamers 2020. Thank you.
Dream Chemistry Award is a contest unlike any other in the world. It concerns dreams, and organizing this contest, we pay tribute to all the dreams of humanity that change and drive the world towards a better future. Well, in fact, we pay tribute to us all, because we are all dreamers. I am a notorious dreamer. When I was 10 years old, I was exposed to the first physics classes, and I told my mom, I will be a physicist. And bingo, 20 years later, I had a theoretical physics group and a permanent position in the Institute of Physical Chemistry. On the blackboard that you see is my second dream that I had at the age of 23. I wanted to better understand the non-equilibrium thermodynamics, and 34 years later, I wrote it on a green board. Along the way, okay, patience is not my second name. Okay, I had to learn to be patient. If your dream can be fulfilled in a matter of weeks, it's not a good dream. It has to take years. So I learned patience, but along the way, I also learned that patience is not enough. When I was 33, and as you remember, I ran the theoretical physics group, somebody showed me a cartoon of an interior of a living cell, and I was really amazed. Yeah, it's so crowded by biomolecules. It's like a Tokyo metro in a rush hour. What you can do in a Tokyo metro in a rush hour, you can just stand, you cannot move, and these proteins move and perform the functions. So I ask myself a question: how do they move? How fast do they move? How to describe it? And it took me 8 years. Well, along the way, I realized that theoretical physics is not enough for biology. So I kept thinking, this is important. You keep thinking and you look for opportunity. And opportunity arrived, I seize it, and I got my first money to solve this problem from the Unilever company. I simply told them, look, I can determine the nanoviscosity of your products. Okay, I was thinking about the proteins of the size of nanometers moving inside a liquid, so of course, nanoviscosity. And interior of a cell is a complex liquid, so in vitro studies, I could use the Unilever compounds. And they simply gave me money. Like, you know, it was my shortest grant proposal, just one sentence. Okay, that's not enough. You have to share your dreams with others. You have to share it with your friends. You have to share it with your colleagues. In this way, you strengthen your commitment. But also, you see, if you make your individual dream, the bigger the dream, the more people has to believe in it. You have to make it a collective dream. If you make it a collective dream, you will see the results that will amaze you, like this one. An individual dream of a first pharaoh became a collective dream of Egyptian civilization, and we still see the results of this dream. You know, ask yourself why Elon Musk is telling everyone that he wants to send people to Mars. You know, why? Because he needs people and he needs money. So he needs to make his individual dream a collective dream. And by the way, don't row against the flow. Elon Musk did not start with dreaming about sending people to Mars. First, he got money. Okay, first he started with a PayPal or something like that. Well, dreams of today tell us something about the future. So we also organized a contest to know the future of science. The future of science is hidden in the dreams of young scientists. And so what we do, basically, we organize the dream, we learn about the dreams, some of them may become a collective dreams, and this will show us the future. All of these dreams that you will know after these finals deserve the statue. You know, the statue, now open your artistic minds. The statue represents a dream. As far as I remember, my first dreams were really vague. I could not pinpoint the right scientific question. So you see, it is, look at the statue. Here you see the vague dream. It's like a vapor, it's highly volatile. Finally, it becomes a droplet, like a first drop of a distillate. You know, a first drop of a distillate. And then you can touch it, you can feel it. And if you make a good stuff in your distillation process, you can even feel the warmth of success inside. So let's start the contest and let's see the future. I will start with one technical remark to the viewers on YouTube. You can also ask questions to our finalists. Simply put your name, your email, the question, and the name of the finalist. And now let's start the contest with the first presentation of Yunyan Q from the Northwestern University. Yunyan, please share your dream with us.
Okay, let me share my screen here. Is it clear?
Yes, we can hear you and we see your slides.
Great. All right. Thank you. So good morning, good afternoon, everyone, depending on which time zone you're right now in the world. And thanks for tuning in to listen to this final of Dream Chemistry Award 2020. So my name is Yunyan Q. I'm currently a postdoc at Northwestern University, supervised by Sir Fraser Stoddard. So I'm really privileged and honored to be able to be selected by the committee to give this presentation about one of my dreams. I hope it's, well, it's the first. I hope it's not the last. So it's about how to achieve the holy grail of polymer synthesis using catalytic artificial molecular machines. So this dream is really based on my experiences in my PhD as a polymer chemist at Carnegie Mellon University, as well as at Northwestern University, and start to work on artificial molecular machines. So let's begin. First, I would like to introduce myself a little bit. My scientific journey, well, started, firstly, I was born at 1988 at the northern part of China, at the border of China and actually North Korea. And then I might find my way to Peking University, one of the best universities in China, and continue my study in pharmaceutical science. I got my bachelor degrees in 2011. And then I decided to continue my chemistry training and move to Carnegie Mellon universities here in the US, at Pittsburgh, and start to work on polymer chemistry. And in 2016, I joined Sir Fraser Stoddard's group at Northwestern University working on artificial molecular machines. And in particular, I'm very interested in investigating the directed motion and polymer synthesis using artificial molecular pumps. I'm super grateful to all the supervisors and great mentors at CMU and NU, especially my postdoc supervisor, Fraser Stutter, who gave me 100% freedom to explore artificial molecular machines, my PhD advisors, Kevin Noonan and Tomek Kowalowski, who lead me into the world of polymer chemistry. I also like to thank my committee members, Krzysztof Matiaszewski and Rong Chao. Chris is also the board of this Dream Chemistry Award, so I see a connection there. So I'm really glad that I have all these people supporting me. I'll also like to thank all the funding agencies, Northwestern University and the ASF, Army offices, Carnegie Mellon University, PRF, and CAST for the supporting of our research throughout my scientific career. So my PhD group, I mainly focus on polymer synthesis and the methodology development. So I'm being particularly involved in studying the transport properties of new materials, in particularly try to design catalytic systems and the catalysts and monomers for control synthesis conjugated polymers. We're also working on gas transport such as CO2 separation membranes using olefin metathesis to afford functional materials, as well as using control radical polymerizations to synthesize ion transport membrane materials that can be used in fuel cell membranes. In detail, I'm particularly interested in using a carbon-based plating catalyst to initiate a steady catalyzed, steady cross-coupling to make poly-3-hexylthiophene in a chain growth polymerization fashion. Then we're able to afford a very high molecular weight of a P3HD. Another thing we're particularly interested is to expand the monomer scope that using a mild Suzuki catalyst transfer polycondensation to make conjugated polymers that incorporating base-sensitive groups such as ester. We'll also start to investigating expanding the scope of monomer. Instead of using thiophene-based conjugated polymers, we're expanding to furan-based conjugated polymers. Then we use Kumada coupling reactions to afford them in a controlled fashion. And perhaps the most relevant work that to this talk is actually we're working on conjugated sequence control synthesis of conjugated polymers by using a predetermined monomer sequences, such as in this case, a thiophene-thiophene-selenophene, a thiophene-selenophene-selenophene, and furan-thiophene-selenophene sequences. The resultant polymers has a perfect periodical sequences of thiophene-thiophene-selenophene and as well as furan. So all these sequence control synthesis give rise to unique properties in their conducting behaviors. A little bit about my postdoc work. So since I joined the Fraser's group, I've been working on how to control the unidirectional motion of molecules, in this case, a ring-shaped molecules, by using artificial molecular pumps. By threading two molecular pumps in a head-to-tail fashion, we're able to power the unidirectional motion of a ring-shaped molecule from the left to the right by two redox cycles. We are also able to couple the redox stimuli as well as the light stimuli that power the unidirectional motion of a ring molecule through the orthogonal stimuli of both redox and light. Perhaps the most important work during my postdoc at Northwestern is to develop a precise polyotaxane synthesizer. In this case, we attach two molecular pumps at both ends of a polymer chain, in this case, a PEG chain. by applying sequentially or in one pot redox cycles, we're able to make precise polyuretaxanes with complete control over the number of rings. We're able to make ring incorporation up to 10, the number of 10. So that's a total of 40, more than 40 charges on a single polymer chain. So the operation of this molecular pump is quite complicated. I won't have the time to explain it. But however, a cartoon will probably tell this in a more accurate or easier fashion. We're using radical pairing actions to power the motion of the ring-shaped molecules. Once upon reduction, the ring got recruited onto the molecular pumps. Upon oxidation, will push inward onto the polymer collecting chain. So this year, 2020, marks the 100 years anniversary since Professor Starginger first coined the term of polymerization in 1920. So since then, polymer chemistry has been exploded to especially the use of the development of commodity polymers, such as automobile tires, insulating materials, or packaging materials. All this development in polymer chemistry has basically revolutionized the everyday life of human beings. So in recent half a century, polymer chemists are really interested in developing chain growth, living polymerization methods that can afford precision polymers. In precision polymer synthesis, where polymer chemists are able to control the length, the composition, the topology of resulting polymers. With all these advances in precision polymer synthesis, polymer chemists can use these materials to assemble various architectures, such as complex micellization, or even organic, inorganic hybrid materials, or non-covalent networks. So all these intriguing complex architecture resulted from the ability to produce all these polymers in a precise way. So in recent decades, polymer chemistry are really at the forefront of making a lifelike living polymers. We know that DNA are genetic codes of human lives. The sequence embedded in the DNA very much defines the life itself. So polymer chemists start to thinking, can we engineer the similar level of sequence controlled polymer, but using synthetic methods, conventional or unconventional synthetic methods that eventually mimicking lifelike living polymers. In this case, the ultimate goal is, can we make artificial synthetic life? So nature has been a good teacher in this case in producing sequence-defined biomicromolecules in a very precise way, such as in DNA synthesis, DNA polymerases are able to recognize and thread onto a growing polymer chain as well as polymerizing the incoming monomer. Similarly, in protein synthesis, ribosome is capable of polymerizing a sequence-defined protein in a peptide chain by threading onto a messenger RNA molecules. In this case, all the biomolecular-based polymer synthesizer, they offer outstanding sequence control, but they are very limited in terms of structure diversities. There's only a handful of monomers that can be incorporated and compatible with this kind of molecular machines. In conventional polymer synthesis, there are two major categories of polymerization reactions. One is the step growth polymerization. So there's no difference between the monomers a reaction between the monomers and the dimers or tetramers. So the reaction reacted randomly. So this results in a very broad polydispersity. A typical step growth polymerizations, such as conventional polycondensations, such as polyester synthesis, polyamide, urethane synthesis. Some advanced step growth polymerizations right now very popular are acyclic diamine methacid polymerizations or click polymerizations. In comparison, in recent half-century, chain growth polymerization has really gained really popular in the polymer synthesis as it can afford well-defined polymer structures in terms of length and composition. There are many types of chain growth polymerizations. In particular, the popular one will be ionic polymerization, radical-based polymerization, cationic polymerization, ring opening polymerization, and ring opening metathesis polymerizations. In recent 20 years, that ATRP and RAFT are probably the most used radical-based polymerization techniques. Chain growth polymerization gave rise to a variety of defined chain length, composition, and the topologies. Now, the real question is, the next generation of advanced materials are probably determined by these precisely ordered monomers. The question is how we can turn the randomly distributed monomer sequences to precisely ordered monomer sequences. Overall, all the chain growth, conventional step growth polymerizations, they offer outstanding structure diversity, but very limited in terms of sequence control. So the polymer chemist field are really working towards how to design catalytic systems that can give rise to precisely ordered monomer sequences. So why we're interested in sequence control polymers? As long been regarded the holy grail of polymer synthesis, we know that sequence-defined polymers are really useful in data storage, as DNA has been the storage medium for human genetic information for millions of years, billions of years. Sequence-defined peptide and proteins can fold and self-assemble into specific structures such as fiber, tube, ring, cage, or other complex structures. These all resulted from a sequence-defined polymer design. Perhaps by studying the folding of self-assembly of these sequence-defined polymers, we eventually will have a better understanding of origin of life. Sequence-defined polymers has also a very great influence on the microscopic properties of resulting polymers, such as in this case, a randomly co-polymerized polyesters actually has a very nonlinear hydrolysis profiles because of the heterogeneous hydrolysis nature. However, a perfectly alternating polyesters give rise to a perfectly linear hydrolysis profiles. This can give rise to targeted on-demand chemical recyclings, may actually solve the problem of plastic pollutions. So there are many ways that currently can afford, in some degree, a sequence-defined polymers, such as in the typical step growth copolymerizations by using the XY type of bifunctional polymerization reactions. You can either afford a statistical copolymers or using the XXYY type of bifunctional monomers, you can afford a perfect alternating copolymers. Similarly to my PhD work using a predefined monomer sequence you're able to get a perfectly defined periodic copolymers. Perhaps the most sequence-controlled way of making sequence control polymers are using a sequential oligomerization reactions. In this way, you are using a partially protected or temporarily protected monomers. Then by attaching the initiator onto a solid support, you're able to make a perfect sequence-defined oligomers or even polymers. But this requires tedious separation, washing, and purification steps. In chain growth copolymerizations, depending on the reaction ratio, the reactivity of A-type or B-type monomers, you can either get gradient copolymers or a alternating copolymers. By learning from nature that by looking back to the DNA replication, DNA synthesis using DNA polymerases or peptide synthesis using ribosome, we found they have shared common scenarios, which they both utilize a chemical reactivity under nano confinement. In this case, both propagating chain and the monomers are confined into a specific environment, in this case under nano confinement, which give rise to the specific sequence control. Now the biggest question, the $1 million question is, can we design such type of artificial enzymes or artificial molecular machines that mimicking the function of biomolecular machines, but using a wholly synthetic way? Since I joined the Fraser's group in 2016, actually right after he got the Nobel Award for the design and synthesis of molecular machines, I have been thinking how we can use artificial molecular machines, which is directly resembling of biomolecular molecular machines in nature, to use all the artificial molecular machines in polymer synthesis. As a synthetic chemist, where either we can make nanocars that can move on the surface, we can also make a molecular size synthetic robot that can do catalysis or make small molecules. But my particular interest is how we can use artificial molecular machines to make polymers, specifically sequence-defined polymers. So artificial molecular machines are, the majority of the artificial molecular machines are based on mechanical interlocked molecules. The two major types of mechanical interlocked molecules are rotaxanes, which are a ring-shaped molecule threaded onto a dumbbell-shaped molecules, or cannonines, where two rings are interlocked with each other. So the synthesis of both rotaxane and cannonines can be achieved by template-directed synthesis. In this case, a donor-acceptor type of synthesis. The blue ring in the cartoon representing electron-deficient ring-shaped molecules, and the red thread or the red ring representing electron-rich molecules. So by donor-acceptor interactions, they are able to assemble with each other. So the definition of a mechanical bond is the entanglement in space between two or more components, such as they cannot be separated without breaking or distorting chemical bonds. One of the most efficient synthetic routes to make MIMs, mechanical interlock molecules, are based on active template synthesis. In this case, macrocyclic ring-shaped molecules can pre-organize two dumbbell components and subsequently catalyze the covalent bonding formation and forming a rotaxane. In this case, for example, you have two dumbbell components that based on using a microcyclic pyridine-based ligand, they're able to form a rotaxane molecules in a very high yielding. So the copper catalyst here pre-organize the two incoming dumbbell components as well as catalyzing the final triazole formation. So my dream chemistry up until now in the short term is, can we transform a step growth polymerization into a chain growth polymerization by using a catalytic artificial molecular machines? So in typical step growth polymerizations, there is no reactivity difference between the monomers and the oligomers or even polymers. However, once the catalysts are mechanically interlocked to the growing polymer chains, the catalyst cannot diffuse back to the reaction medium, therefore serving as an initiator as well as a catalyst. And only incoming monomers can be grown onto the propagating chains. In this way, it can promote a chain growth polymerization. This is quite reminiscent to my PhD study on the catalyst transfer polycondensations in the synthesis conjugate polymers. So for example, in a typical catalytic cycles for cross-coupling reactions, if you use palladium tetracase phosphine or palladium DBA, some normal palladium-based catalyst, there's no reactivity difference between the monomers or dimers, and the monomers, and the tetramers. So you result with a randomly distributed polymer. In this case, a conjugated polymers. So you will have a very broad polydispersity. However, if you use a very electron-rich phosphine-based ligand, such as the bidentate nickel DPPP, DPPE, or T-butyl-phosphine ligand, actually, after each propagating event, the metal catalyst will coordinate it to the growing polymer chains and never falling off from the growing polymer chain. It forms a metal polymer complex in this case. So after each propagating event, the catalyst will transfer to the terminal of the growing polymer chains and catalyze a new propagating new monomer to be incorporated to the polymer chain end. In this case, it's called a catalyst transfer polycondensation. So the strength of the metal polymer pi complex between the metal and the polymer backbones are very important in determining how regulated, how controlled this chain growth, this catalyst transfer polycondensation can be. So my design using a microcyclic ligand will prevent the catalyst to ever falling off the chain because of mechanical interlock nature of this catalyst. So in detail in the particular catalytic transfer polycondensation, after the reductive elimination of the cross-coupling reactions, if the nickel will fall off of the product, the diffuse back to the reaction medium, it will promote a step growth polymerization. But in comparison, in reality, it will actually form a metal pi complex, and this will promote a chain growth polymerization. So this nickel pi complex will further oxidative addition intramolecularly to the terminal CBR bond. And the incoming monomer will transmethylate and reductive eliminate and forming the trimer, but with the catalyst still attached to the polymer chain end. So the propagating event continues, and eventually you're getting a well-defined poly-3-hexylthiophene in this case, with a dispersity well below 1.5. So in my design, the monomer can be a click type AB monomers, in this case with an amide bond in between. So this resembles a peptide surrogate, so it actually have biological properties. The catalyst, we can use a bipyridine-based copper catalyst. So the proposed mechanism starts from using an initiator with a bulky end group. So under basic conditions, the copper catalyst will thread to the end of this initiator, forming a sigma-bound copper acetylide complex. The incoming monomer will coordinate to the copper catalyst inside the cavity of the microcyclic ligand. Through 6-member ring cycle addition reactions and subsequently forming a triazole product. And this propagating event can be continued until you reach a certain amount of polymers. However, in this case, the catalyst never diffuses back to the reaction medium, thus promoting a chain growth polymerization. So literature already has evidences towards this kind of mechanisms. So Professor Gao in 2015 actually reported this hyperbranched polymer synthesis based on click polymerizations. In this ABC type of click monomers, instead of step growth polymerizations, they propose that the copper catalyst never leaves these hyperbranched polymers, that they are all coordinated to the polytriazole propagating chains, therefore forming a hyperbranched polymers in a chain growth fashion. So in 2018, Professor Goldav also reported using similar catalytic active template synthesis to make catenines. Pay attention to this kind of AABB type of monomers. Instead of making a 3-catenine or 4-catenine, they actually only are able to isolate the 2 catenines. Which in this case, that means the catalyst must catalyzing one triazole ring and subsequently transfer to another side and catalyze the subsequent triazole formations. No matter there are 2 triazoles or even 4 triazoles. That means the catalysts are able to transfer throughout the formation of these newly formed rings. So in this case, I can call this a catalyst transfer click reactions. This gives evidence that my proposed synthetic routes will work. So in further design, we can also synthesize a carbon-based microcyclic ligands. In this case, the metal can be various. You can use palladium or nickel or ruthenium. So this really opens up many possibilities in the carbon design. We can also, in this case, if we're using a ruthenium-based carbon precatalyst, we are probably able to regulate the acyclic diamine metathesis polymerizations, similarly as the click reactions. The key throughout the mechanism is based on there's no chance that the catalyst can diffuse and fall off from the growing polymer chains, thus promoting a chain growth polymerization reactions. So in the long term, I'm hoping the current research efforts are able to expand in various directions, including how we can increase the complexity of our microcyclic ligands. In this case, can we incorporate multi-site catalytic pockets within the microcycles, such as A, B, C, D, 4 types of catalytic sites? We can either use metal catalyst or organoid catalyst, which has been proved in the literature that can catalyze a certain type of polycondensation reactions. We can even increase the complexity by making a 3-dimensional molecular cage-shaped molecules. In this case, we can incorporate multiple sites, even more than just 4, but also taking advantage of this complicated nano confinement effect. The recent years has been witnessed great development in nanopore sensing technologies. My question is, by combining my design of artificial catalytic molecular machines, by designing a specific type of proteins that resembles a nanopore, by incorporating multi-reaction functionality inside the pore, are we able to make sequence-defined polymers in a more controlled fashions? We can not, we don't need to be bound to the biological-based nanopore sensing technologies. We can either use the solid-state-based or hybrid-based nanopore sensing technologies. My goal is, can we functionalize the inner channel of the nanopore with multi-site catalytic catalysts? In this way, we can not only power the polymerization by tuning the voltage, then depending on the different charge state of the incoming monomers, we can screen out different type of monomers to be incorporated into the resulting polymers. We can also use the current to monitor and determine which the monomer is actually, in fact, incorporated into the polymer structure. And finally, I think recent years there is a great development in de novo protein engineering techniques. As protein engineers, they are able to make specifically computer predicted peptide structures that can self-assemble into predicted structures. So by using de novo protein engineering, are we able to make a complex complex bio nanopores that actually self-assemble into the structure that we need, and also incorporating the catalytic reaction sites that we desired. So in summary, my dream project is really the combination, based on the combination of precision polymer synthesis and the use of artificial molecular machines. Can we use, so in essence, we want to transform a step growth polymerization into a chain growth polymerization by mechanically interlocked catalytic artificial molecular machines. And ultimately, can we design a complex artificial molecular machines that can mimic the function and achieve the complexity of biomolecular machines? So all these efforts might eventually lead to a better understanding of origin of life, as well as the higher possibility to make artificial synthetic life. So finally, I would like to thank my postdoc supervisor, Fraser Starter, who gave me the 100% freedom at Northwestern University to explore the chemistry that I like the most. Also, I would like to thank the great group we have here at Northwestern. We have numerous collaborations that we are free to discuss our dream chemistry projects. Also, I would like to thank Robert, who really put a lot of effort in this contest, and also the people working behind the scene at organizing this Zoom meeting and the YouTube broadcast. And lastly, I would really thank the Dream Chemistry Award Committee for giving me the opportunity to present my dream chemistry here. Thank you.
Thank you, Yunyan. The first question is from Daniel Gryko. Daniel, please.
that was interesting dream. But like for the dream, which by definition is new, you are, as a main tool, you are using the reaction, the click chemistry, developed by Sharpless and 14, which is sort of worn out. I mean, so many people are using this cycloaddition for a variety of different purposes, which becomes, I think the word worn out is quite relevant. So can you imagine another, let's say, interaction, not copper triple bond and not something analogous, which would give you the same effect chemically?
Yes. So, yeah, you are right. Actually, the reason I choose the click chemistry is because of the, as you said, it's broadly used in polymer chemistry or in other chemistry field. So click chemistry possesses probably the most broad scope of monomer functional group. So in this case, if you're able to use click chemistry that in a chain growth fashion, you will actually start to afford a variety of different functional polymers by incorporating, because click chemistry is the most, probably the most versatile chemistry there, chemical reactions there. However, you're right. We are also, as I mentioned later, the next part of exploring the catalytic artificial molecular machine is by designing other type of ligands. I think carbon-based ligands are among the most interesting type of ligand, because carbons has been widely used in palladium, nickel, ruthenium-based chemistries. The use of palladium, nickel, or ruthenium metals really opens up many possibilities, such as in cross-coupling reactions, in ring opening, in metathesis reactions, in variety of different kind of reactions. You can also, I didn't show other designs. You can also make a silane type of ligand, but using a microcyclic design. So in that case, you can coordinating to titanium, zirconium, other type of transition metals. So that also opens up many possibilities for other type of chemical reactions. Yeah.
I think, maybe other people also would like to ask the question. Thank you very much. Good answer.
Thank you.
Hey guys, I think that we used up the time for the questions. We will have a 10 minutes break before the next presentation. So 10 minutes break and we'll be back with Claudia Bonfire. So see you in 10 minutes. To change the world.
Dream Chemistry Award is a unique contest in which a prize is awarded to a young scientist with a chemistry background for an idea of a scientific project in the field of chemistry or chemistry-related disciplines that she or he dreams to solve.
It's a unique competition in the sense that we don't reward achievement, we reward chemical dreams. And what I really like about it is that it's targeted to young rising stars, not to the Nobel Prize winners, but to those who may get the Nobel Prize in 10, 20 years.
With this prize, we want to strengthen the will to pursue the goal in science, to pursue the dream. And we strongly hope that it will change the world.
In 2017, the competition, organised by the leading chemistry institutes of the Czech and Polish Academies of Sciences, saw a record number of nominations of promising scientific talents from around the world. During the final in Prague, 5 finalists presented to the International Scientific Committee projects ranging from targeting the surface of tissue cancer cells, engineering the microbiome in the gut as a tool for metabolism applications, developing microelectronics based on metal organic frameworks, or optimizing electron transfer reactions, to ending mercury pollution in gold mining.
The winner is Jessica Kramer, and she came with a beautiful project on the extracellular matrix, glycocalyx, and its role in cancer. And this is a potential for completely new view of addressing, investigating cancer and potentially treating cancer by actually addressing the fact that cancer cells have a different sugar coating around themselves, and we can use it as a target for therapy. It's a very bold project. It's very far from being realized. It's not clear whether this will ever be therapeutically useful, but it's certainly interesting, it's exciting, and I think it's worth awarding the prize.
So this competition is very unique and amazing because usually you win awards for the work you've already done. And this competition is very special because you win an award for a dream, for an idea that you have not yet accomplished. And so, you know, part of dreams are motivating you to go out and do new science and accomplish wild, interesting things. And so it's really amazing to have an award that supports the future.
Światło to jest niesamowite zjawisko. To jest podstawowy składnik materii całego wszechświata, który tak naprawdę spaja wszystkie atomy, molekuły, wszystko co znamy. Światło definiuje czas i przestrzeń. Jest zupełnie niesamowitym czynnikiem, który my jako ludzie możemy odbierać bezpośrednio za pomocą naszego wzroku. Obdarzeni jesteśmy możliwością kontroli światła, możliwością sterowania tym światłem i używania go, na przykład do obrazowania, do tego, żeby w komputerach mieć obraz naszej tkanki. Tym właśnie zajmuję się ja i mój zespół, próbując oswoić światło i zastosować je do czegoś praktycznego.
W naszej grupie zebraliśmy specjalistów z bardzo różnych dziedzin, więc z łatwością zajmujemy się zagadnieniami z pogranicza biologii, medycyny, farmakologii czy fizyki laserów.
Wszyscy moi znajomi uważają mnie za pewnego rodzaju magika i poniekąd jest to prawda, bo w tej pracy łączymy różne elementy takiej zaawansowanej techniki optycznej, wyrafinowanej obróbki danych, do tego szczypta inżynierii, żeby tworzyć urządzenia, które poniekąd są magiczne, bo pomagają diagnozować choroby różnego rodzaju, które normalnie nie są widoczne gołym okiem.
Nauka obok sztuki jest jednym z najważniejszych aspektów działalności człowieka. Sztuka może leczyć duszę, ale nauka może leczyć ciało. My zaczynamy od oka.
Lasery, modulatory, mikroskopy i inne urządzenia to jest dla mnie fascynująca zabawa. Eksperymenty naukowe, mimo że często są bardzo zaawansowane, są do ogarnięcia przez jednego człowieka. Teraz pracuję nad taką zaawansowaną techniką obrazowania ludzkiego oka. Mam nadzieję, że ta technika będzie kiedyś stosowana przez lekarzy, wspomagając wczesne wykrywanie różnych schorzeń w oku i będzie techniką nieinwazyjną, to znaczy jak najbardziej bezpieczną dla pacjentów.
I like building microscopes, but as any other instrument, it has its own limitations. So my job is to improve the performance of a microscope. This also means that what I'm doing day to day hasn't been attempted before, which could sometimes be challenging, but always exciting.
My dreams are to finish my project, which is really very challenging, to study about the small growth of the tumors and do something to understand the basic biological and physical process beyond this, to understand how they're really developing and have like a good pharmaceutical drug to cure it.
Pracuję nad nowymi metodami obrazowania, które zrewolucjonizują te metody, które mamy obecnie w mikroskopii, czy też generalnie w kamerach.
Fajnie współpracować z ludźmi, dla których rzeczy niemożliwe nie istnieją. Myślę, że najlepsze słowa opisujące pracę u nas w zespole to mierz wysoko i sięgaj celu.
My research aims to better understand how human eyes work. To me, being a scientist is having the opportunity and the beauty at the same time of learning something new every day.
W naszych projektach cały czas staramy się rozwijać nowe metody, a jednym z nadal nierozwiązanych problemów jest problem, jak zobaczyć komórki pod skórą, jak zobaczyć komórki w żywym organizmie. Naszą ambicją jest rozwiązać ten problem. Jesteśmy blisko tego, widzimy już światełko w tunelu, jesteśmy coraz bliżej, ale to jest początek drogi.
Welcome back after the break. The next dreamer is Claudia Bonfire, University of Cambridge. Claudia, the Zoom is yours. Share your dream with us, please.
Can you see it fine?
Yes, all fine.
Okay, great. So I'll start with saying that so first of all, good evening, everyone, and good morning to those of you that are on the other side of the ocean. I am very excited to be virtually here with you tonight. I would have loved to be there. But I'm very excited to be here and have the opportunity to share with you my chemistry dream. To do that, we need to take a few steps back in time to about 4 billion years ago, when our planet was rather different and life was not yet here. How did life start on Earth? There can hardly be a more intriguing question. Some of you might think that as we cannot go back in time, we might never know exactly what happened. This is maybe why Albert Eschenmoser said that the origin of life cannot be discovered. The origin of life has to be reinvented. Now, we might not be able to go back in time yet, but we can explore the biochemistry that exists right now and reinvent a 4 billion years old biochemistry that existed back then. Actually, in the last 20 years, we've seen huge advances in our understanding of how life could have emerged on early Earth and how it could be reinvented. We have now clues for the synthesis of life's ingredients, for their combination into small active proteins, for their elongation into short replicating nucleic acids, and for their assembly into membrane-like structures. Now, I like to think that I have contributed a bit to our understanding of how life's building blocks have emerged and how they could have been combined together in the same way we would make things out of Legos. During my PhD with Sherif Menzi in Italy, I looked at primitive catalysts, combining amino acids with metal ions. Modern life is heavily dependent on metal ions, and approximately half of all proteins are metalloproteins. Among metal-based cofactors found in biology, iron-sulfur clusters constitute one of the most ancient, ubiquitous, and structurally diverse classes of biological prosthetic groups. But how did iron-sulfur proteins emerge on early Earth, and when they started to play such an important role for extant life? During my PhD, I've discovered the synthetic pathway for the formation of prebiotic iron-sulfur clusters driven by UV light that can be considered the most abundant source of energy present on early Earth. And these prebiotic iron-sulfur clusters can sustain multiple cycles of redox reactions, transfer electrons between biologically relevant substrates, and generate a proton gradient across lipid membranes. Alongside catalysis, replication of nucleic acids is a critical process for the emergence of cellular life. Repeated steps of annealing and separation of complementary RNA strands is required to achieve multiple cycles of non-enzymatic RNA replication. As a postdoc in John Sutherland's group in Cambridge, we've shown how the melting temperature of RNA double strands can be modulated by small variations in pH, enabling the separation of these RNA double strands at very mild conditions and potentially supporting multiple rounds of non-enzymatic RNA replication. Now, in modern life, the cellular content is kept together by highly heterogeneous lipid membranes. Primitive membranes were likely very heterogeneous as well, but they were probably formed of much simpler components. So how did early membranes evolve into modern membranes? Still as a postdoc in Jan Sutherland's group, I proposed a synthetic strategy for the emergence of self-assembling lipids, prebiotic lipids, that share some features with modern lipids and that could selectively accumulate over non-self-assembling molecules through energy dissipative cycles. Now, the next step after having worked on all of these life's building blocks was to combine everything together to explore the potential coexistence of life's ingredients. So as a Marie Curie Fellow in Jan Sutherland's group, I look at the compatibility of early membranes with potential chemistries occurring within them. And not only we found that the activation of amino acids and nucleotides to yield peptides and oligonucleotides was not by the presence of lipid membranes, but also the same chemistry synergistically yields novel species which are capable of localizing to the membrane, inducing their growth and driving the interaction with other biomolecules. All of these components, if combined together, can form what's called a primitive cell or protocell that can be ideally considered the ancestor of the oldest living system. The last few years have also seen important advances in exploring how primitive cells could grow, replicate themselves, and divide. Actually, during the first edition of the Dream Chemistry Award, you chose as the winner a visionary colleague of mine, Evan Sproit, that I hope is here with us tonight, who dreamt about artificial cells that could do exactly this: grow, replicate, and divide. This is a recent video from Evan Sproit's research group that shows, for example, how coacervates, that are simple artificial cells, can incorporate each other, you'll see it here, can incorporate each other and be hierarchically organized into multiple coexisting domains. Another contribution exploring the dynamic behavior of protocells comes from Sherif Menzi's group, where protocells, and you can see them here, are shown to divide into smaller protocells upon the addition of an external trigger. In the last few months, I have also started to look into what protocells do when exposed to temperature gradients. What you can see in this video are lipid protocells that, when the temperature rises, the phase transition into oil droplets. You'll see this in a second. And these oil droplets, you'll see, start to coalesce into bigger oil droplets. And the aqueous content of these protocells is entirely released in solution. When the temperature starts to decrease, these oil droplets become very unstable. You'll see in a second that they'll start to move around, they'll become very dynamic, and eventually they'll start to form daughter protocells budding from the oil-water interface. So overall, these three videos that I've shown you show that primitive cells had a very dynamic and vibrant life on early Earth. So going back to what Albert Eschen-Moser said and what I've shown you at the beginning about the origin of life and the fact that we can reinvent it, we can reinvent the chemistry that led to life's ingredients, and we are doing it. We can reinvent the chemistry that led to dynamic primitive cells, and we've just started to do it. But this is not yet enough. These protocells are still missing one of the most fundamental lifelike behaviors. These protocells are missing the ability to communicate. This is why my dream is to reinvent primitive communication, that same communication that emerged about 4 billion years ago between very simple primitive cells, and that allowed them to efficiently and continuously sustain the prebiotic chemical processes that were hosted within them. So my dream is to reinvent primitive communication and build primitive cells that can talk to each other. Why is that? Well, communication is the first skill that we learn in life. We start interacting with the world outside by crying when we are babies, first to start breathing, and then to ask for food, for sleep, or for cuddles. We grow and we keep interacting constantly and sometimes even unconsciously with everything that is around us. We learn how to send a message and we learn how to receive it. We learn how to talk and we learn how to listen. We learn how to express ourselves through verbal communication and nonverbal communication. Tonight, we are via meetings and YouTube channels. We cannot stop receiving messages from the outside, not even if we close our eyes and we are in a silent room. But communication is a skill that goes beyond human interactions. For example, there are certain squids and octopuses that can change the color of their skin to selectively interact with each other. For example, to attract females and to repel other males. Bees release pheromones to interact with each other and to manage colony organization. Maple trees, when they are under attack, maple trees send airborne signals to the neighboring maple trees to warn them of the impending danger, so that the other maple trees around them can start pumping out bug-repelling chemicals. Communication is a skill that belongs also to the microscopic scale. And at the microscopic level, the language is chemical and the messages are molecules. For example, neurons communicate through a chemical process that involves the release of a neurotransmitter and its binding to the receptors of a target cell. Organoids. In this picture, you can see brain organoids that are miniaturized and simplified models of our brain. Brain organoids can interact with the media in which they are grown and selectively uptake nutrients and drugs. In eukaryotic cells, there's a tight communication network that has evolved between mitochondria and the nucleus, including signaling cascades and sensing of mitochondrial products by nuclear proteins. Quorum sensing is also a process of cell-to-cell communication. It allows bacteria to share information with each other about cell density and adjust gene expression. So all of these examples that I've shown you show how communication is ubiquitous, universal, and defining property of living communities, both at the macroscopic and the microscopic level. We and our communication systems have just been around for a few 100,000 years. Simpler organisms like bacteria have been around for much longer, almost 4 billion years. And with them, also their ability to exchange chemical messages. So why did this start? When did this start? And how did this start? Well, modern cells are self-sustaining units of life. However, they do not live in isolation. They interact with each other and with the world outside constantly. To do so, they exploit complex biochemical machineries to release and uptake chemical messages that are needed for the chemical processes that they host to occur. So how could primitive cells, 4 billion years ago, in the absence of those complex biochemical machineries, exchange chemical messages? How could they, chemically speaking, talk? We've just watched videos of primitive cells growing, dividing, and physically interacting with other protocells. Those primitive cells have then the potential to interact with each other and with the world outside. They have the potential to create vibrant and dynamic communities of primitive cells. They have the potential to communicate. So could they? Could they communicate? Nobody has yet found an answer to this question, and that's why this is my dream. So how can we do that? How can we reinvent protocellular communication? Well, let's think about this together. What are the requirements needed to communicate? For example, let's say that tomorrow you want to have a coffee with a friend that lives in a different. What do you need to communicate with your friend? Well, it turns out that it's pretty simple. Communication only has 3 requirements. Clearly, you and your friend, the message that you want to share with your friend, and a tool that can mediate the transmission of the message. So what would this mean in protocellular terms? What are the minimal physical and chemical requirements for primitive communication? Well, first, protocellular communication would require a message, which would be molecules produced through prebiotic chemical processes. Then protocells, that are the prebiotic compartments that can host these prebiotic chemical processes. And then biochemical tools that can mediate the molecular transmission process. So how can these 3 requirements be combined together? How can we make my dream come true and reinvent protocellular communication? Well, I think we would need 3 steps. First, we need to build communities of different protocells in order to understand if they are capable of coexisting. Then we need to take each of those protocells and understand if they can support exchange of chemicals with the environment. And then we can move up in the complexity scale and combine communities of responsive protocells and build protocellular communication networks. So let's think about each of these steps a bit more in detail. To support protocellular communication, primitive cells would need to be compatible with each other. But can protocells coexist? Can they collaborate? Can they compete? Do they kill each other? Just to give you an idea about what kind of molecules we are talking about, these are some of the prebiotically plausible lipids that can be employed to build primitive membranes. So what happens if you build 2 different populations of primitive cells and then mix them together? Well, when 2 populations of phospholipid protocells, that here are reported in red and green, are mixed together, as you can see here, they peacefully exist, nothing happens. But this is not always the case. When fatty acid protocells are mixed together, they merge and a new population of yellow protocells forms. So you see, identifying populations of protocells that can coexist is fundamental to understand if they can communicate. Second step, communication with the environment. We would need to understand how protocells, each different protocell, can support the uptake, the release, and the exchange of chemical messages with the environment. To do so, we would need to explore the prebiotic chemical space to find molecular systems, what I was calling earlier biochemical tools, that can functionalize primitive membranes and make them responsive. So once they are functionalized, can primitive cells communicate? Responsive primitive membranes could have interacted with the outside world in several different ways. For example, mediating the selective recruitment of molecules to the surface of the membrane, or supporting their catalytic conversion, supporting and sustaining transport and signal translocation across the membrane, or molecular interaction. But all of these activities result from the functionalization of the lipid membrane. I have already shown in the past how exploiting a prebiotic systems chemistry approach, relevant biomolecules could be localized to early membranes. And this type of membrane functionalization drives membrane growth, co-localization of interesting biomolecules to the surface, or their catalytic conversion. So building on these findings, more advanced functionalities could be explored. Communication with the environment would mostly be based on signal and solute translocation across the membrane. But what kind of membrane functionalization can support this? Again, I just want to show you a bit what I'm thinking about. So I'm thinking about two classes of compounds. First, combined systems made of lipids and either nucleotides or amino acids. And these molecules would be capable of localizing to the membrane and participate to or catalyze chemical reactions in response to environmental stimuli. A second class of molecules we can think about are amphiphilic peptides. These can dynamically interact with primitive membranes in response to external triggers. Both of these biochemical tools have the potential to drive the interaction between primitive cells and their environment. So diving a bit into some details and some ideas about the chemistry behind my dream, to achieve signal translocation, I can think about two strategies. First, we could design prebiotic anchors that can be switched between an inactive and an active state by an external signal. For this, I'm taking inspiration from my PhD work on iron sulfur clusters, and I'm thinking about systems in which we have a lipophilic anchor so that they can interact with the lipid membrane, and a metal complex. And as we've seen during my PhD, that UV light can be used to convert iron 2 to iron 3, and so convert reduced iron complexes into oxidized iron complexes, we can think about designing prebiotic photoresponsive anchors so that we would have the inactive reduced complex anchored to the membrane. And then by switching on the light, we would convert the reduced form into the oxidized form. And this would then be able to catalyze chemical reactions. A second example is to design prebiotic molecular transducers. This takes inspiration from the seminal work of Chris Hendon at the University of Cambridge. And for this, the idea would be to design systems that are made of 3 different modules. You can see them here in green, in gray, and in blue. The intermediate one, the middle one, would be a lipophilic spacer so that this system can interact with, this module can interact with the membrane. And then there would be 2 switches on each of the ends of the transducer. So the way this system would work is that we would have the transducer sitting on the membrane and exposing one of its modules. This would be a hydrophilic polar modules. And when it interacts with something that we have in the outside of our protocell, it gets converted into a hydrophobic module. This would push the transducer across the membrane and expose the other module, the other end of the transducer, that would then be able to interact with something that is in the internal of our protocell, in the inside of our protocell, and then be converted into an active form that can again catalyze chemical reactions. For the solid translocation part, again, I can think about 2 different systems. I can think about prebiotic carriers and prebiotic channels. In this case, we can take inspiration from naturally occurring peptide-based systems that are capable of transporting small molecules and metal ions across the membrane. And we can think about designing a library of peptides that have similar properties to these naturally occurring peptides, and yet are made of prebiotic amino acids. Once we have this library of peptides, what we can think about is to select some of them that are capable of transporting small molecules and metal ions across the membrane in an active and controlled way. This can be achieved, for example, by using peptides that can switch between a closed and an open form in response to external signals. For example, upon the formation and the disruption of salt bridges by pH or ionic strength changes, or the formation and the disruption of disulfide bonds upon the addition of a reducing agent. At this point, we would be ready to combine these two steps and explore communication between protocells. Once we have protocells that are compatible with each other, and each of these protocells is capable to interact and communicate with the environment, it would be possible to combine communities of coexisting protocells and achieve protocell-to-protocell reciprocal communication. This could be done in a symmetrical way with different protocells that have similar membranes functionalization, or in an asymmetrical way with different protocells that have different membrane functionalization. And as a proof of concept, we can think about combining communicating protocells that host redox processes. For example, one would host a reduction reaction and the other one an oxidation reaction, and they would just exchange the reactants and the products, or ester formation and hydrolysis. But we can also think about more complex communication systems that would employ, for example, non-enzymatic RNA replication or self-assembling systems. Overall, the realization of my dream will enable us to build networks of protocells of higher complexity that exhibit complex lifelike behaviors and that could support cascade reactions and molecular and supramolecular oscillatory patterns. So in this past 26 minutes, I have told you about my dream. It's based upon and how I could possibly make it come true. You hopefully felt that I am really excited about my dream. But why should you be excited with me? Well, first, because building protocellular modules whose activity can be switched on and off and finely modulated can help to develop novel responsive sensors and materials. For example, glucose-responsive liposomes or photosynthetic liposomes. Second, because identifying the key features and functionalities that led to protocellular communication can help to design minimal artificial supramolecular assemblies with lifelike behaviors. And third, because reinventing protocellular communication can bring us one step closer to connect chemistry and life as we know it. I like to think that research in the origin of life field is like the game Connect the Dots. We have the dots, we know what the picture should look like, we just need to connect them in the right way and in the right order. So how can we connect the dots between chemistry and life? Well, first thing, dream big. We can dream big, and in fact, we should dream big. Then, we just need to find the number one and start connecting the dots. Thank you.
Thank you, Claudia. It was really inspiring. And I see that the first question is from Pavel Jungwirst. Pavel, please.
As follows. So you're trying to reinvent the primitive communication, and this is an important part of the, say, origin of life story. And when people talk about this origin of life, they always talk about emerging phenomena. So what kind of emerging phenomena would you expect in your system? Something you didn't put in and should come out? I mean, do you have any concrete ideas where to look for? What to expect? Thank you.
Yeah, so it's a very interesting question. And what I like about this field is that there are always a lot of questions, and it's nice to invest time to find the answers. So what I'm thinking about are both sort of short and long terms goals. So something that we can think about, for example, is to explore peptide synthesis. So now we've just seen a couple of papers in the last few years published by Matthew Pauner's group that developed a way to achieve peptide synthesis in a prebiotic way. Something that would be very interesting, for example, is to explore whether peptide synthesis, prebiotic peptide synthesis in the presence of lipid membranes can yield some of these prebiotic channels or prebiotic carriers that I've discussed about, so that the combination of this chemical process and this supramolecular assembly can yield peptides that localize to the membrane and favor the transport. Something else that we can think about, for example, and I hope I'm answering your question, is if, for example, we have some chemical reactions that produce molecules that then can have a specific behavior, for example, self-assemble into other systems. So I'm thinking about, for example, decoacervates that we were seeing in the first videos. If we can find ways to uptake the nutrients from the outside and inside have a chemical reaction that form more complex molecules, more complex peptides that can self-assemble into coacervates, then we can have some kind of advantage in having this whole system, so this sort of communication with the environment, because we can uptake a molecule, we can uptake the starting material, and then we can have a whole new phenomenon happening inside the vesicle, the protocell.
Thank you very much.
The next question is from Daniel.
Somewhere in the middle of the lecture, you describe these basic aspects of communication: the sender, the receiver, and the message, and the means. And you describe them, almost all of them, in a chemical way. Maybe I missed something, but except for the actual message. So, no, the message only in your cartoon was coffee. So what would be the message here? What they communicate about?
So, in a very, very simple term, when I was practicing this talk with a friend, actually he asked exactly the same question. So, clearly, we can think about these messages as being simple molecules. So we are mostly discussing about transport of material, let's say, from one cell to another, or from the outside to the inside. But something we can think about is also what I was mentioning when I discussed about quorum sensing. So we know that quorum sensing, it allows bacteria to communicate with each other about cell density, for example. So we can think about a situation, a system in which we have primitive cells that are communicating a sort of information, a sort of a sort of information about either the density that they have in solution, or some other kind of phenomena or chemical reactions that are occurring. So that's more or less what I'm thinking. It's easier to think about practical things, so molecules moving from one side to the other, but I think there is a lot of that can be explored in terms of information as well.
Thank you.
You're welcome.
Okay. The next question is from Jacek Waluk. Jacek, please.
For this exciting talk. But I was a little bit confused when you identified your message with a molecule. I would rather think that molecule is a tool, not a message. But as a spectroscopist, I have a question. Did you ever consider using light as a tool, which tells you something about the state the sender is in?
Yeah. So it's definitely something that would be worth exploring. Mainly because, as I said, UV light can be considered the most abundant source of energy available on early Earth, and so it would be very interesting to understand how this whole system is affected by switching on and off the light, for example. But I started mentioning when I was discussing about prebiotic photoresponsive anchors, about systems that can be designed by using UV light, for example, that can switch between one state and another. But in general, there isn't much known about the effect of light, also in terms of their impact on these primitive cells. So it's definitely something that should be explored in quite a wide range in general.
Thank you. If I may, just the second one. Why do cells communicate? I think that the main purpose is, of course, to stay alive, right? And now the question comes, would you be able to distinguish between useful and information and not useful information, or you may even call it noise? I think this is quite challenging, but is it doable?
Yeah, so I think what I'm thinking about is that if you think about a primitive cell, we are used to think about primitive cells as containers that host a chemical process. What we tend to think about is that we provide the chemicals, the reactants for this chemical process to occur, and then it would just keep occurring. But at some point, these primitive cells would not be capable anymore to uptake the nutrients. And so at that point, you need the chemistry to be the protocell to be able to communicate with the outside to take more nutrients. Otherwise, the chemical reaction will just stop occurring. So that's why I think that communication is fundamental.
Okay. The next question is from Janusz Bujnicki. Janusz, please.
Which are related to each other. The first question is, do you think that it will be easier chemically to introduce a completely new method for the artificial, simple pseudocells to communicate with each other, or to introduce a new method for the existing cells to have another layer of communication, something new that cells do not use? And a related question, do you think that it will be possible to introduce something like social media, so to take existing cells and introduce a new channel of communication that could become addictive, so the cells would start communicating with each other instead of doing something else?
That's a really interesting question, a couple of questions. I think it would be really nice, and I don't think we should in general constrain ourselves by what we know that happens, for example, now in our cells. So clearly we are inspired and we look at what biology does in order to either design our systems or to analyze, because it's easier for us to recognize what we know rather than what we don't, right? But at the same time, I don't think we should constrain ourselves too much, especially because when I was mentioning the kind of applications that one could get out of this whole dream, then of course, if we can find new ways of communicating or new systems that can allow this communication to occur, then that would be definitely very useful and potentially really interesting for other kind of artificial systems. So I don't think we should constrain ourselves at all.
But what would be easier on the chemical level?
So I would say that it's probably easier to recognize what we know. And so if we have a model to look at, if we look at biochemistry and biology, it's probably easier to follow that path rather than recognizing a completely new one. But it would be amazing to find a new one.
Thank you.
Okay, thank you all. And we have a next speaker, our dreamer, Pavel De Dio. So Pavel, now the Zoom is yours. Please share your dream with us.
Thank you, Robert. Thank you so much for organizing it. And thank you so much to all viewers who are here to hear about my organic chemistry dream. And I stress here, organic chemistry. But before I will tell you about my dream, I would like to tell you a few words about myself. So, in fact, I'm Polish and I was educated in Warsaw. So it's a pity that we cannot have this event in Warsaw. And I studied chemistry as a major and biology as a minor. And in that time I joined the laboratory of Professor Janusz Jurczak. And afterwards I moved to Amsterdam to work under the mentorship of Professor Joost Ray, where I received my PhD. And with that experience, I moved to the US, where I was a postdoctoral fellow at UC Berkeley with John Hartwig as an advisor. And in 2016, I came back to Europe, but this time to France, and I accepted the position of an assistant professor at the University of Strasbourg in this beautiful building. Okay, but also a few words about what I've done over the years. So my chemical interests are around organic chemistry, and I was always fascinated by the way nature conducts organic reactions. And of course, those are mostly conducted by enzymes, which are natural catalysts. And here we have an example of, from synthetic organic chemistry point of view, an amazing enzyme which can hydrate this oleic acid with excellent selectivity, which is very difficult to achieve with standard small molecule catalysts. And if we look closer at the active site of this enzyme, we'll notice why such enzymes are so selective. In fact, a substrate is precisely pre-organized in the substrate binding site, and this is highly influenced by the non-covalent interaction between the carboxylic group and amino acid residues. And in fact, when I was in college, I joined the group of Professor Jurczak to study supramolecular chemistry, so chemistry above the molecules. And there, among different projects, for instance, we studied artificial recognition sites, such as this diindolylmethane motif, which turned out to be an excellent receptor for a deprotonated carboxylic acid. But as I said, after my master's, I moved to Amsterdam, and there we asked ourselves a question whether we can use this kind of non-covalent interactions to build up enzyme-like catalysts. Because we know that catalysts are great, however, they do not operate for all kinds of reactions that we have access to as synthetic chemists. So what was our mode of operation? We would take an unselective catalyst for hydroformylation reactions, so reaction of alkenes to produce aldehydes, which is incredibly important in industry, but in many cases, it's very unselective. So we would take this unselective site, we would combine it with a recognition site, which we know that interacts strongly with deprotonated carboxylic acid. And in that way, we can construct a bifunctional enzyme-like catalyst, which has now a catalytically active site and substrate binding site. And we studied this system in detail, and indeed it has an enzyme-like property and can solve many selectivity issues. So for instance, this kind of bifunctional catalyst can conduct very selective reaction on this oleic acid type substrate with very high selectivity, while traditional catalysts perform very poorly in this kind of systems. But when I moved to Berkeley, we asked ourselves a different question, whether instead of building up a new enzyme-like catalyst, we asked ourselves whether we can repurpose a natural enzyme to perform a new reaction. And this work was done in a close collaboration with Yunyan Q, which was a grad student at that point in the group. And what was the logic here? So we would, in this case, take a heme protein, such as myoglobin, as our model platform. And this metalloprotein contains an iron protoporphyrin as a cofactor, which is in the active site. And we postulated that if we swap the iron for a different metal, we can in that way build a new reactivity of such system. And besides introducing a different metal, we can also introduce a different ligand coordinating the metal site, which will modify, fine-tune the properties of such system. And we are very flexible here. So we can introduce different metals, we can introduce different axial ligands, then fine-tune the properties of such constructs, and we can also use different protein scaffolds. And we developed a method to quickly prepare arrays of such catalysts that can be tested for reaction of interest. And this kind of constructs were, for instance, tested for a biological reaction such as carbon insertion reaction into CH bond. And indeed, a construct that contained native iron were not active, while many other constructs were active with the iron containing versions of the enzyme being the most active. Then the question was whether this kind of systems, they're active, but are they selective? Because they did not evolve towards a new reaction, right? In fact, they're very unselective. However, applying a logic of directed evolution, the one proposed by Francis Arnold, we could quickly identify very selective variants of such enzymes. However, the dirty secret here was that this kind of artificial metalloenzymes were not very active. They would perform just merely, let's say, 4 turnovers the most per hour, which is fairly poor comparing to native enzymes. So we redesigned our approach and we took a different scaffold, which is known for being very robust and contain a true substrate binding site. And this kind of system was already much more active. And upon couple of rounds of directed evolution, we could quickly evolve the enzyme towards very high activities, which were comparable to activities of natural enzymes. So this was the first example of artificial metalloenzymes which could perform abiotic reactions with kinetics of native enzymes. And in fact, nowadays, this kind of enzyme can be also incorporated in vivo into E. coli to create artificial metabolic pathway. So, okay, so this is some old results, but now I would like to talk about my true chemistry dream. But before I tell you about my chemistry dream, I would like to ask you, what are the major challenges in chemistry? And as we see here, we have all different dreams. So I guess we all see different challenges, but I would suggest that one of major challenges, in particular in organic chemistry, is low overall efficiency. And I'll try to explain why. So if we see the efficiency of producing materials and chemicals that we use every day, we are not so great. So for instance, while fuels or bulk chemicals are produced with very high efficiency, the fine chemicals require a lot of work and generate a lot of waste and consume a lot of resources and energy and have a very negative impact on our planet. So I think this is one of challenges that we should address if we think about pursuing sustainable society. But why is that? Why production of fine chemicals is encumbered with generation of a lot of waste? So this comes from the fact how we currently conduct synthesis of chemicals. Essentially, we think about synthesis in a linear way. So we start with accessible starting materials, generate intermediates, and iteratively convert them further towards target products. And each step requires resources and also generates waste. So of course, the more complex, the higher level of complexity within our fine chemical, the more steps are required. And that leads to a lot of resources consumption and generation of waste. But don't understand me wrong. Organic chemistry is fantastic and it's extremely powerful. So let's have a look at this molecule. It's called paleotoxin. It's one of the most toxic molecules on Earth. Essentially, often just one molecule like this can kill a single cell. And this kind of molecules are found in coral reefs or organisms living in this area. But from an organic standpoint, this is a highly complex molecule, and it has been called as the Mount Everest of synthetic chemistry. Because you can imagine that to construct such molecule, we need to stitch together many small fragments. And in fact, organic chemistry, as said, is powerful, and the synthesis of paleotoxin has been achieved by Professor Ishii at Harvard, so one of the giants in organic chemistry. However, the synthesis took over 10 years. So let's say that this is quite striking when comparing it with biosynthesis, which is conducted via just this simple organism. So perhaps there is a better way, or there is a different alternative way to conduct organic synthesis. So the question is, how can we improve the efficiency? So yes, we can think about developing new reactions, new selective transformations. But besides that, let's maybe have a look how nature makes this kind of molecules. So here I present you the metabolic networks present in Escherichia coli, very simple bacteria. And you can see that this is a highly complex network that encompasses synthesis of all molecules of life, from lipids, carbohydrates, amino acids, DNA, and so on and so forth, and including also secondary metabolites and natural products. So it's very complex. However, if you closely analyze that network or any biosynthetic network, you will notice that biosynthesis is conducted with just 6 classes of enzymatic reactions. And all reactions occur under ambient conditions. And just the combination of all those reactions, all those 6 classes of enzymatic reactions enable all diversity of natural products. So this is a fairly limited toolkit, especially if we compare it with toolkit of laboratory synthesis, where we have myriads of reactions which do not occur in nature. We have thousands of highly selective catalysts, many transitional metal catalysts that do not occur in nature. Many reactions like cross-coupling, the metathesis of alkenes, those do not occur in nature. However, somehow laboratory synthesis is encumbered with this low efficiency. So I think that there is one significant difference between biosynthesis and laboratory synthesis. So while in laboratory synthesis, each reaction is conducted independently, in biosynthesis, all reactions occur in parallel. And I do think that this is a significant advantage of biosynthesis. So my overall dream would be to enable synthesis of target products via constructions of artificial networks of organic reactions in a way that combine the reactivity of simple starting materials to produce directly target products. You may ask, what's the advantage of conducting everything in parallel? So there are three key features. So in such systems, you can have, the reactions can cooperate with each other. So material or energy of one process can drive another process. So there is no loss, there is no waste, or there are no additional reagents required. And of course, such networks can go through unstable intermediates or highly reactive intermediates, which cannot be accessed otherwise. And here, because they are consumed once formed in subsequent steps, there is such pathways are enabled. And besides these two features, there's one big advantage. Such systems do not require any intermediate isolation and purification. And this is an important factor. So for instance, GlaxoSmithKline estimated that in their synthesis of fine chemicals and pharmaceuticals, just the solvent used, which is typically used for isolation and purification of the intermediate, accounts for over 80% of material that is utilized for processes and for over 75% of energy and big part of the negative ecological input. So my idea is to study such systems so that we can enable direct transformation of starting materials into target products. And how can we do this? How we approach this? So I like the quote from the previous talk. So dream big, but start small. So we start with small systems so that we can understand all interactions within it. So for a second, just forget about networks. Just think as an organic chemist that you need to convert this alcohol to beta functionalized alcohol. And if you just analyze the reactivity of this molecule, you will quickly realize that the position that you want to target is less reactive than the other side, which is activated by the neighboring functional group. So if you subject it to typical reaction conditions, you will rather prepare alpha functionalized product than the targeted beta functionalized product. But then you can think of, okay, so we have a functional group, and if you remember something from organic chemistry, you know that you can use the functional group as a directing group, which will grab a catalyst and bring it close to the site that you want to functionalize. However, in this case, you are also out of luck because the system will rather form a 5-member metallocycle than 4-member metallocycle due to its higher stability. So overall, again, you will form a different product than you wanted. So you are in this unlucky situation when the site that you want to functionalize is neighboring less reactive site. But now we know also that we can modify the reactivity by performing some transformation on the molecule. So for instance, we can oxidize the alcohol to the aldehyde, and then the reactivity pattern completely reverse. So now the site that you want to functionalize is reactive, can undergo easy functionalization, and then you can reduce it aldehyde back to the functionalized alcohol. So it's effective, but it's not very efficient because you need to conduct 3 reactions, and you also use stoichiometric amounts of oxidants and reductants just for changing the reactivity at some point. So can we do all these transformations at once? So in other words, can we use networks of catalytic reactions? So here we thought, okay, we know that alcohols react reversibly with some metal complexes to form aldehyde and actually ruthenium hydride, but in the absence of any hydrogen acceptor, this reaction is reversible, meaning that if you have a lot of alcohol, a little bit of your metal complex, you will always form a little bit of aldehyde reversibly. Now we also know that aldehydes can undergo easy arylation with aryl halide in the presence of palladium catalyst. So we wonder whether just combination of those two reactions into this relatively simple network can connect directly the functionalization of alcohols at the beta site. So again, how does it work? Alcohol is reversibly oxidized to aldehyde, just in small quantity. However, once formed, it undergoes the functionalization to form the functionalized aldehyde, which is also in equilibrium, but now with the target product. And in the end, here, due to the dynamic nature of equilibria involved at those steps, all starting material is pushed forward to form the target product. And note that here we formally do oxidation and reduction. However, we don't use any oxidants or reductants because we couple two opposite steps, formal oxidation, formal reduction. Perhaps more importantly, both reactions occur under mild conditions, so overall reactions should also occur under mild conditions. So in other words, we form this reaction pathway around the high energy barrier for the direct transformation. So this is the idea, but you need to conduct it in the lab. So long story short, David, who is the main player in this in the project on alcohol, he identified quickly that this pair of catalysts is compatible and can conduct this reaction. And here we have those advantages of the networking reaction. So it's very efficient, no oxidant, no reactant, one pot, and reactions occur under mild conditions. And because of that, the method is applicable to a broad scope of substrates, including some bigger molecules. And in principle, this strategy is rather general. So you can think of connecting different reactions and in that way enable new transformations. And this is essentially we study the scope and limitations of this approach within part of our ERC grant project. But I don't want to discuss this. I want to tell you a little bit about consequences of reaction networks. So I told you about connecting those two reactions. So, however, alcohol, which is supposed to react with ruthenium, it can also react with palladium. So for instance, it can undergo O-arylation, or it can undergo oxidation, or even intermediates such as aldehyde and alcohols can reversibly react to form hemiacetals, which is just another type of alcohol. So you would guess that this has some consequences for the network. So perhaps the system is not as simple as initially thought. So let's have a look at this. What is the full reaction network here? So we start here. We start with the alcohol, as before said, oxidized to aldehyde, and which is related to functionalized aldehyde and reduced back to alcohol. But hang on a sec, we have an aldehyde here, which can also again undergo arylation. So we form third type of aldehyde in the system. And this aldehyde can also be hydrogenated back to the alcohol. So then we have three different types of alcohols. The initial one, monoarylated and disarylated. And we know that alcohols can undergo O-arylation. So we have three different ethers. And as said before, we have alcohols and aldehyde, so they can form hemiacetals. That's okay, they are formed reversibly, but those can be reversibly oxidized to the esters. So in that way, just starting with two substrates and two catalysts, we can generate a lot of different products. Just to make it even a little bit more complex, we have three different alcohols. We know that alcohols can also undergo irreversible oxidation. making the system a little bit more complex than initially shown. So in other words, when you want to conduct a direct transformation of alcohol to arylated alcohol, you want to promote those three processes while at the same time suppressing all other processes. And it has a consequence on thinking about this kind of systems. So for instance, at this case, you do not need to use the most active palladium arylation catalyst, but you want to use the most selective catalyst, which does not promote any other reactions if you are interested in this product. So that's okay. But within this network, we can form many different products. So can we also control the selectivity here? So what I mean, can we design the input parameters and that employ on the network the selective formation of one target outcome? So in other words, can we select suitable catalysts and conditions and starting material to drive the network towards selective formation of one of possible products beyond the initially designed? So how do we think about that? So for instance, let's consider that you would like to directly convert alcohol, these unfunctionalized alcohols, to beta-related aldehydes. And if you carefully analyze the topology of this network, you will note that there are three possible pathways that you can end up forming this member of the network selectively. So those are noted with red color, blue color, and green color. And of course, all of them are different, but they share some similarities. So for instance, here, all of them need to promote both arylation steps of the arylation of 2 and 3. And at some point, they also need to promote selective oxidation of alcohol, whether this is unfunctionalized, monofunctionalized, or dysfunctionalized. And at the same time, all other processes need to be inhibited. And if you consider another member within the network, such as this bisarylated alcohol, the situation is a little bit more strict. You need to promote very selective arylations and very fast hydrogen borrowing while inhibiting all other side reactions. So we wonder whether we can somehow systematize this kind of analysis. So we noted that we have 4 classes of products. So we have initial alcohol, alcohol ether, ester, and aldehyde, and those can be also functionalized to a different level. And now, if we think about conditions that promote formation of each of those classes, we can see some patterns. So in other words, we can construct this kind of decision tree of conditions that will ensure selective formation of each class of the product. And we can see that the formation of alcohols over ethers or over oxidation products is mostly controlled by inherent properties or selectivity and activity of the catalyst, while, for instance, the type of oxidation products, so whether it's ester or aldehyde, is largely controlled by the concentration of the reaction. And of course, the level where we end up, whether this is monofunctionalized, dysfunctionalized, or triple functionalized reagent, the material depends on the initial conditions, initial ratio between the starting material. So with using this kind of topological analysis, we can identify suitable catalysts for each targeted product. So in that way, we selected suitable conditions, and we can, within the same network of reactions, we can form different products with high selectivity for each class. But of course, we don't need to stop here. We can go farther. We can introduce another level, another dimension. So we, instead of just using two substrates, we can add another substrate. In that way, further functionalize the one starting material, the one intermediate. And this is quite amazing for me, that just starting with 3 substrates, 2 catalysts, we can quickly access a relatively wide chemical space of material. Okay, so I showed you our bottom-up approach, where we focus our analysis on the actual network. So the careful topological analysis enables to identify rules which promote formation of certain products. So this is
1 minute left.
Okay.
Okay.
Okay. Yeah. So I'll go faster. So it's a rational, but it's not quantitative. So we cannot directly connect the we cannot directly build models which define the outcome of the network. So for that, instead of focusing on the network, maybe we should directly connect the input with output. So study the influence of the starting conditions on the outcome. And this, in that way, we have direct correlations and direct experimental correlations. So we can see the global analysis of influence of each parameter. However, those data are highly complex because the system itself is complex, and such effects are difficult to read. And so what we think now, that how to address those issues, we can implement here machine learning, which enables to uncover the correlations within complex sets of data with subtle effects. And in that way, we can build quantitative models which drive the development of networks toward the selective outcomes. But in the end, our end game is not to study the networks. We want to provide tools which enable to develop with the aim, with keeping the target product in mind. We want to enable development of networks which enable the connection of accessible starting materials and transformation them to target products. And in principle, we can think of using chemoinformatics tools for that, where we can implement rules for the different reactivities, and driven by the chemical intuition, we can propose the reactions that we can imagine that could lead to the target product. Then with such chemical generators of reactivity, we can implement algorithms that build directly full networks of reactions by exploiting the full connectivity. And subsequently, with topological analysis supported with machine learning, we would like to find suitable conditions and the elements of the network towards formation of the target product. And in the overall dream would be to enable in that way the formation of compounds, not only in a more efficient way that they are accessed now, but also chemicals that cannot be accessed nowadays with a classic stepwise approach. And with that, I would like to acknowledge my group and some funding, and would like to thank you for your kind attention.
Okay, thank you, Pavel. One question comes to my mind when I see this network of reactions. How you would synchronize them, because they compete for common substrates. So if you want to work all the network in concerto, you should think about synchronization. How would it look like in your scheme?
Should compete. They all should occur at the same time. You just need to select certain elements. So even within, because what's the key is the selection of a catalyst, right? Because, and by the selection of catalyst, the important part is the implementation of a good ligand or a suitable ligand. So for instance, we can control within this network that we have, by the size of the ligand, we can control substrate selectivity. That the same catalyst can perform, for instance, hydrogenation on one substrate, on smaller substrate, but it cannot conduct hydrogenation or oxidation of the bigger substrate. And the same counts for our relation. We can control via the just chemical properties of ligands. But so so what we do currently, we do it experimentally, and then we try to correlate this. But we would like to perhaps have a precise description of this kind of the parameters of the kind of ligand, and we see directly how they reflect the properties within, within or in isolated case or within the network.
Okay, thank you, Pavel. The next question is from Daniel Gryko, please.
Look, as we both know, even for relatively simple organic transformations, and even these days, there is still very painful and long-term optimization. You know, we can argue that one day it will finish, but it's not coming soon. Even the best papers from the best groups, from the best universities, there is still table of optimization at the very beginning. And the reason for that is that we are not really able to compute it yet. So this is complex enough. And then with this complexity of your networks, and of course it's becoming even more complex. So my point is that you obviously have to really heavily optimize it. And so do you plan to do it by some sort of robot? Because it would take years, you know, to find the right solvent mixture, catalyst, all these parameters.
So essentially, so what I showed, so first of all, yes, it's painful, but I think because often people don't think about side reactions and they do it just, you know, empirically, right?
Yeah.
And that's what in our initial study, that's what we did. And we couldn't optimize it very well, right? We optimized it for, you know, 70%, we were happy. But now with understanding of the full system, we can go above, you know, 90% because now we know what exactly matters there. So we can then back engineer it, right? We apply this knowledge now to the new transformation. So there are limitations to the topological analysis because all those parameters are intertwined, right? And now the idea is we exploit, we find those complementary, like the topological analysis with machine learning, that we want to also see those subtle effects and understand them in a greater detail.
That answers the question. Thanks a lot.
Okay, thank you very much. And now we will have a 10 minutes break. So see you in 10 minutes.
Biochemistry Award is a unique contest in which a prize is awarded to a young scientist with a chemistry background for an idea of a scientific project in the field of chemistry or chemistry-related disciplines that she or he dreams to solve.
It's a unique competition in the sense that we don't reward achievement, we reward chemical dreams. And what I really like about it is that it's targeted to young, rising stars, not to the Nobel Prize winners, but to those who may get the Nobel Prize in 10, 20 years.
With this prize, we want to strengthen the will to pursue the goal in science, to pursue the dream. And we strongly hope that it will change the world.
In 2017, the competition, organised by the leading chemistry institutes of the Czech and Polish Academies of Sciences, saw a record number of nominations of promising scientific talents from around the world. During the final in Prague, 5 finalists presented to the International Scientific Committee projects ranging from targeting the surface of tissue cancer cells, engineering the microbiome in the gut as a tool for metabolism applications, developing microelectronics based on metal organic frameworks, or optimizing electron transfer reactions, to ending mercury pollution in gold mining.
The winner is Jessica Kramer, and she came with a beautiful project on the extracellular matrix, glycocalyx, and its role in cancer. And this is a potential for completely new view of addressing, investigating cancer and potentially treating cancer by actually addressing the fact that cancer cells have a different sugar coating around themselves, and we can use it as a target for therapy. It's a very bold project. It's very far from being realized. It's not clear whether this will ever be therapeutically useful, but it's certainly interesting, it's exciting, and I think it's worth awarding the prize.
So this competition is very unique and amazing because usually you win awards for the work you've already done. And this competition is very special because you win an award for a dream, for an idea that you have not yet accomplished. And so, you know, part of dreams are motivating you to go out and do new science and accomplish wild, interesting things. And so it's really amazing to have an award that supports the future.
Światło to jest niesamowite zjawisko. To jest podstawowy składnik materii całego wszechświata, który tak naprawdę spaja wszystkie atomy, molekuły, wszystko co znamy. Światło definiuje czas i przestrzeń. Jest zupełnie niesamowitym czynnikiem, który my jako ludzie możemy odbierać bezpośrednio za pomocą naszego wzroku. Obdarzeni jesteśmy możliwością kontroli światła, możliwością sterowania tym światłem i używania go, na przykład do obrazowania, do tego, żeby w komputerach mieć obraz naszej tkanki. Tym właśnie zajmuję się ja i mój zespół, próbując oswoić światło i zastosować je do czegoś praktycznego.
W naszej grupie zebraliśmy specjalistów z bardzo różnych dziedzin, więc z łatwością zajmujemy się zagadnieniami z pogranicza biologii, medycyny, farmakologii czy fizyki laserów.
Wszyscy moi znajomi uważają mnie za pewnego rodzaju magika i poniekąd jest to prawda, bo w tej pracy łączymy różne elementy takiej zaawansowanej techniki optycznej, wyrafinowanej obróbki danych, do tego szczypta inżynierii, żeby tworzyć urządzenia, które poniekąd są magiczne, bo pomagają diagnozować choroby różnego rodzaju, które normalnie nie są widoczne gołym okiem.
Nauka obok sztuki jest jednym z najważniejszych aspektów działalności człowieka. Sztuka może leczyć duszę, ale nauka może leczyć ciało. My zaczynamy od oka.
Lasery, modulatory, mikroskopy i inne urządzenia to jest dla mnie fascynująca zabawa. Eksperymenty naukowe, mimo że często są bardzo zaawansowane, są do ogarnięcia przez jednego człowieka. Teraz pracuję nad taką zaawansowaną techniką obrazowania ludzkiego oka. Mam nadzieję, że ta technika będzie kiedyś stosowana przez lekarzy, wspomagając wczesne wykrywanie różnych schorzeń wokół i będzie techniką nieinwazyjną, to znaczy jak najbardziej bezpieczną dla pacjentów.
I like building microscopes, but as any other instrument, it has its own limitations. So my job is to improve the performance of a microscope. This also means that what I'm doing day to day hasn't been attempted before, which could sometimes be challenging, but always exciting.
My dreams are to finish my project, which is really very challenging, to study about the small growth of the tumors and do something to understand the basic biological and physical process beyond this, to understand how they are really developing and have like a good pharmaceutical drug to cure it.
Pracuję nad nowymi metodami obrazowania, które zrewolucjonizują te metody, które mamy obecnie w mikroskopii, czy też generalnie kamerach.
Fajnie eksperymentować z ludźmi, dla których rzeczy niemożliwe nie istnieją. Myślę, że najlepsze słowa opisujące pracę u nas w zespole to mierz wysoko i sięgaj celu.
My research aims to better understand how human eyes work. To me, being a scientist is having the opportunity and the duty at the same time of learning something new every day.
W naszych projektach cały czas staramy się rozwijać nowe metody, a jednym z nadal nierozwiązanych problemów jest problem, jak zobaczyć komórki pod skórą, jak zobaczyć komórki w żywym organizmie. Naszą ambicją jest rozwiązać ten problem. Jesteśmy blisko tego, widzimy już światełko w tunelu, jesteśmy coraz bliżej, ale to jest początek drogi.
Welcome back after the break, and now, Ivana, the Zoom is yours. Please share your dream with us.
Thank you, Robert. Good evening, ladies and gentlemen, or I guess I should say good day to all of those joining from different time zones. Firstly, I would really like to take this opportunity to thank Robert and, of course, all the organizers of the Dream Chemistry effort for this event, which gives young researchers this unique opportunity to think about their dreams and share their dreams in maybe some sort of different format what we are usually used to. Although with a little bit of a bittersweet taste of not being able to do this in person, I feel really honored to be chosen among this inspiring group of people and present my dream chemistry. However, before I get to talking about my dream chemistry, let me first a little bit introduce myself. So as my first name or last name, let's say, may suggest, I was born in Slovakia. This beautiful country in the heart of Europe, in a small town in the south called Velký Katíš. And growing up, I fell in love with natural sciences, mostly influenced and inspired by my godmother, who is a terrific biology and chemistry teacher. So when the time came to actually choose what I would like to do as my career and study at the university, the choice was quite clear. I decided to combine the biology and chemistry and to study biochemistry at Masaryk University in Brno, Czech Republic, where I stayed for both my bachelor and master degree. And as soon as I started, I actually got very keen on to also already start working in the research lab. And I was very lucky to get accepted to work in Lakshmi Laboratories, one of the European leading groups in biocatalysis. Here, I focus on the characterization of a special class of enzymes called haloalkane dehalogenases, and actually for identification of their members in a special organism called extremophilic organisms, which should potentially give them this enzyme various extreme properties. This research undeniably sparked my love for enzymes. And I dreamed about continuing and traveling the world and going to all these extreme environments to collect this microorganism on my own. However, at the end of my master degree, I also felt that I'm not really done learning. And there is so much more science to still take a look into or explore. So I decided to switch a little bit and move to Netherlands and continue with a PhD in organic chemistry at the Strating Institute for Chemistry at the University of Groningen with Professor Gerard Rufus. Here I spent 5 wonderful years working on a number of different projects, which primarily focus on the development of different methods for designing of artificial metalloenzymes, which culminated in this very pretty PhD thesis. During my time at the Strating Institute, I felt I definitely gained a lot of new scientific dreams, as I felt very inspired by the environment filled with talented researchers from all over the world, and as well, of course, this institute being a home of a Nobel Prize laureate. At the end of my PhD, I also wanted to kind of explore more of applied research. So I decided to move to Graz University of Technology to join a big industrial project which focused on the discovery of novel enzymes towards production of bioplastics from renewable resources. And this was a project I really enjoyed. And working on this project, to having to think in a little bit of a bigger scale than what we usually do in academia, definitely pushed me to start thinking about some of my chemistry dream in different, more applied perspectives. Finally, a couple months ago, I started my new position at the Free University of Amsterdam as an assistant professor, where I hope to continue bringing my scientific dreams to reality. So after this introduction, I guess it probably comes to you as no surprise that a lot of my scientific dreams involve enzymes. The dream I have chosen to write my Dream Chemistry Award proposal actually deals with the special area of enzyme research, and those are enzymes in challenging often new to nature reactions. And more specifically today, I will be talking about enzymes phosphorylation reaction. But first, let's take a step back and start with enzyme. Why are enzymes interesting and why should we even care? Enzymes are biomolecules which are vital to all forms of life. They are micromachines which have been developed over millions of years to catalyze countless chemical reactions in and outside of the cell. And they are remarkable catalysts. They make and break bonds with unmatched rate acceleration and impressive selectivities. And given their abilities, of course, they have a lot of potential to be explored in industry. In some applications, for example, like fermentation, enzymes have been used for hundreds of years. But nowadays, we can also see their widespread application, for example, as detergents in the preparation of biofuels, or for example, in the preparation of biopharmaceuticals. And the global market of enzymes is exponentially growing. Very importantly, enzyme represents green, environmentally friendly alternatives to many chemical processes. As they work in mild conditions, they definitely use less or no hazardous or toxic chemicals. They, as we already mentioned, have high activities and selectivities. They are biodegradable, and we can produce them from renewable resources. Therefore, their use definitely fulfills majority of the parameters for a greener and sustainable future. So obviously, enzymes have a lot of great properties. But at this point, one can of course ask, are there even any drawbacks to work with enzymes? And several issues which have been previously connected with working with enzymes, as for example, low thermal stability or stability in organic solvents, are nowadays being routinely solved. However, one major drawback of enzymes still remains, and that is their narrow reaction scope. Especially if we think in comparison to chemists and what chemists can do, what type of the chemistries they can access, enzymes can definitely do just a part of that. And if we want to apply enzymes in all areas of lives, in all industries, we are actually standing in this very important, formidable challenge. And those are enzymes which have the ability to catalyze all types of biological transformations. So being able to prepare those enzymes or develop the methods which kind of shows us how to develop those enzymes is definitely a big scientific dream of mine and, well, of many others. So one of the challenging chemistries I am particularly interested in is fluorination reaction. Why fluorination reaction? What we know is about fluorine is that it is a key element in the synthesis of molecules broadly used in medicine, agriculture, and materials. Fluorine's properties, such as electronegativity, size, lipophilicity, dramatically influence properties of compounds which contain fluorine. And then these compounds have very different binding affinities, bioavailabilities, and other pharmacokinetic properties in comparison to their non-fluorinated variants. Therefore, we can see fluorine actually being a part of more than 30% of pharmaceutical actually on the market, having fluorine in the structure. And here there are many of the most profitable drugs of all times. For example, as Lipitor, which is used for the treatment of high cholesterol, and it used to be one of the most prescribed drugs ever. So high demand for fluorinated compounds definitely prompted scientists to develop a large number of methods for fluorination chemistry. And this still remains to be a very active field. However, as typical for many of the chemical methods, the chemist ways of how to do the fluorination reaction comes with harsh conditions, toxic reagents. Very often they have to use high catalyst loading, and we often see a poor selectivity outcome, for example. Those efficient and mild methods for the fluorination reaction are highly desirable. So if we turn to nature for solution, which is believed to be one of the best chemists, we realize that actually Mother Nature is not so great in fluorination chemistry, since biochemical processes for bond-forming reactions for fluorine-carbon bond formation are rare. Only 5 unique natural organofluorine compounds are known, and then several more of their modifications. And they are, for example, mostly identified as a toxic tropical plant metabolites. The major reason behind why actually fluorine is so rare in the nature, or there is so little of the organofluorine compounds available, is that fluorine is not really easily available. It's really its low bioavailability, as most of the fluorine is actually bound in insoluble form. Furthermore, its high redox potential prevents typical activity of haloperoxidases, which are used for many biochemical processes for other halogens. Furthermore, a high solvatation of fluorine in water also represents a big obstacle for using fluorine as nucleophile. Therefore, actually, biochemical mechanisms of how these organofluorine compounds have been produced remain largely unsolved and undiscovered. In 2002, it was the time where a first enzyme for fluorination reaction was actually characterized. Discovered in the gram-positive bacteria called Streptomyces cattleyi. And here this enzyme actually is known to mediate a reaction in between the fluoride ion and S-adenosyl L-methionine to generate fluorodeoxyadenosine, utilizing SN2 mechanism and unique dissolution strategy for fluoride ion. Several homologous enzymes have been identified ever since, and in the, let's say, past 17 years from the other bacterial species. However, these are all enzymes which exhibit the same mechanism as the first identified enzyme and the same very narrow reaction scope. Although significant efforts have been used to study this enzyme and engineer it to maybe potentially accept different substrate, this have not proven very successful, and this extremely narrow reaction scope, extremely narrow substrate scope prevents the further kind of applications of these fluorinase enzymes in biotechnology. So identifying new fluorinases therefore continue to be of high interest. Of course, there is still plenty to be discovered in nature. As I have already said, there are enzymes which should be responsible for the fluoroacetate production in higher plants, which have been shown to be present in South Africa or Australia. But these enzymes still remains to be identified and characterized. No one knows how this fluoroacetate production is done in these higher plants yet. Furthermore, it is believed that new fluorinated entities and fluorometabolites are still to be discovered, and therefore new enzymes may appear. However, next to the discovery of natural fluorinases, what I dream about is actually to take the vast knowledge of the fluorination chemistries we have from the chemist and bring those into the biological world. So what do I mean by this? A promising strategy to discover or design new enzyme with the new to nature functionalities, as such could be the fluorination reaction, utilizes the knowledge of chemical or biochemical reaction mechanisms in order to identify enzymes which would share similar mechanistic elements with our desired new to nature reaction. Upon the identification of these so-called promiscuous catalytic activities, what we can do is we can mimic the natural evolution, which we can see in nature, and we can evolve these initial low-level activities into highly efficient catalysts with the powerful methods of protein engineering, such as directed evolution. The prime example of this strategy actually built on the identification of promiscuous activities in heme-binding enzymes, such as here mentioned cytochrome P450. In nature, this class of enzyme is identified in numerous metabolic processes and is known, for example, to activate molecular oxygen and catalyze impressive range of difficult transformations. However, as have been demonstrated in the lab of Professor Francis Arnold, this enzyme actually demonstrates remarkable promiscuous activities as they allow for the formation of the new intermediate species, carbene or nitrines, to be activated instead of molecular oxygen. And this strategy has been shown to be has a lot of potential in a lot of different challenging chemical transformations, such as cyclopropanation, azidation, energy insertion, or for example, creation of carbon silicon or carbon boron bonds, which have been previously not described in the nature at all. Therefore, when I started to think about my dream of actually designing a fluorinase enzyme, I was immediately curious whether cytochrome P450 or other heme-binding enzyme could do this. Could we somehow identify their promiscuous activities as a solution to do this job? I took a little bit look into the literature to identify whether this was already previously done, maybe with the iron porphyrines. And unfortunately, I was not able to find this type of examples. However, what I came across was actually that in 2012, chemists showed that they were able to redirect the hydroxylation activities of manganese porphyrines and use them for fluorination reaction in quite challenging chemistries in the direct fluorination of unactivated alkanes. What was actually very interesting about these papers was that these manganese porphyrines were initially prepared for the hydroxylation as a mimics of P450 enzymes, which led me to believe that maybe exploring heme-containing enzyme may potentially lead to promiscuous fluorination activities. However, transition method catalysis is not the sole inspiration for installing promiscuous reactivities. Another very interesting example involves tautomerase enzyme called 4-oxalocrotonate tautomerase, which contains a rare catalytic feature, N-terminal proline. In nature, this N-terminal proline actually acts as a general base and promotes a ketoenol tautomerization in the catabolic pathway of aromatic hydrocarbons. However, inspired by the versatility of a small molecular organocatalyst, this enzyme actually has been shown to catalyze a variety of different carbon-carbon bond formation reactions, such as Michael addition, aldol condensation, or recently a cross-aldol reaction via formation of an amine or iminium ion intermediates. Given possibilities of N-terminal proline to act as a general base, general acid, or a nucleophile, it is believed that a vast majority of the chemistry which could be shown for these promiscuous activities, which could be shown for these enzymes, remains to be identified. Therefore, if I thought about whether this type of the promiscuous activities could be somehow turned into a fluorination reaction, or could we make this tautomerase into a fluorinase, the inspiration from chemistry here comes from Macmillan group, who reported L-proline and imidazoline catalysts, which are able to catalyze alpha fluorination reaction of aldehyde via an amine intermediate. Similar to previous example, we see kind of mechanistic similarities in reaction intermediates, which represents those kind of important starting point where we could start and explore the potential of 4OD for this reaction. Admittedly, I have significantly simplified chemistries of these examples. And of course, many issues could potentially arise, for example, like with oxo-transfer reagents or fluorination reagents, which are generally used in these chemistry approaches, not being compatible with the protein's environment and would have to be optimized. However, I still consider these two enzyme classes to be a great starting point in order to identify potential artificial fluorinase. However, although of course, several other examples could still be discussed with natural proteins. These are not only two classes where I could see a potential for fluorinase, but for my third and final example, how I would maybe make a dream of artificial fluorinase to come true, I decided to step a little bit beyond what natural enzyme can provide us with. And I actually got an inspiration for this idea when I was still exploring the previous idea. And that was when I looked at the catalysts, which were organocatalysts, which were able to do the fluorination reaction. And here we have a simple building block, a proline, which was shown to be an OKID catalyst in the previous fluorination reaction. And we have this imidazolidinone-based catalyst, which were being significantly better. And this partially reminded me of enzymes, and maybe why we have some problems with enzymes is that actually enzymes are built from these 20 simple building blocks, out of which only 10 can provide some kind of catalytic functionalities. So maybe that could be the reason why they are having troubles to access the challenging reactivities. However, nowadays, we are actually not limited to only these 20 natural building blocks. And we are able to actually use genetically encoded unnatural amino acids, which can, in theory, have any desired modification in its side chain. And upon this modification, we can introduce these unnatural amino acids into an appropriate biomolecular scaffold, giving us new enzymes, new artificial enzymes with desired new functionalities. So in the case of my dream artificial fluorinase, we potentially could kind of turn this known small organocatalyst into a natural amino acid, and upon the appropriate assembly, we can then obtain artificial fluorinase with new functional residues. Of course, this is, as in theory, the unnatural amino acids can have any desired modification. So here we are not only limited to only these organocatalysts, and we can actually explore any other functionalities, functional moieties, which have been used to accelerate or enable fluorination reaction. And upon the then incorporation into the biomolecular scaffold, achieve novel fluorinases. So identifying and prepare. In this process, we can be quite universal, and we can achieve, in theory, any enzyme for any challenging reaction. So identifying and preparing this type of unnatural amino acids is currently a central focus on my group at the Free University of Amsterdam. So in conclusion of my today's dream talk, I hope we can all agree on the fact that the enzymes are a perfect catalyst, and the use of enzyme catalysis in the wide range of industrial application represent an essential effort and approach towards sustainable future. Furthermore, given our current possibilities to combine our natural resources, so natural enzyme, with the modern engineering tools or biological building blocks, I'm confident that we are only mere steps away from building a new robust artificial fluorinase. Finally, I would like to thank you all for your attention, and I will be happy to answer any of your questions.
Okay, thank you. Thank you, Ivana. The first question is from Janusz Bujnicki. Janusz, please.
Thank you very much. Great talk. My first question is, what would be your favorite application? In which protein would you like to put the artificial amino acid? To do what? That cannot be done now.
So in general, for unnatural amino acids, definitely I am very keen on exploring different challenging reactivities. That's why I like to describe a fluorination reaction, because it's definitely challenging and there are so few fluorinases known. So for me, a fluorination is kind of a final thing where what I would really like to approach. However, knowing that it's a very challenging reaction, so that may not be possible. But any other challenging reactivities, I think there's many which can be explored with unnatural amino acids.
But which exactly reaction? I mean, which protein? How would you rearrange the nature to do something with chemistry that nature hasn't invented yet, but could be done with your chemistry?
Perfect. Okay, now I get it. Well, it really depends here when you are talking about different enzymes. When we are incorporating natural amino acids, we are usually not actually looking into already existing enzymes, but we are taking biomolecular scaffolds, which often do not have any previous reactivities, but they are for example, very good for binding. We introduce the unnatural amino acids into those, and then we can kind of explore chemistries with these new functionalities. Those are also then easily kind of engineered further, because you don't have that already existing enzymatic activities which you would potentially compete with or have to get rid of. So we try to kind of explore a different biomolecular scaffold with kind of a different binding properties. What I could mention here, what I was thinking about when I was preparing this presentation, is definitely maybe potentially part of this biomolecular scaffold could be something which could help with the dissolution of the fluoride ion, if we want to go through that chemistry. So building something similar, which is already known in fluorinases, but introducing that into maybe a kind of designer protein scaffolds.
Thank you.
Okay, there's something that I could not grasp in your presentation. Why fluorine is used in so many pharmaceuticals, while in nature it is not used that often? So what's so special about the fluorine that people, probably by trial and error, started to put it into the compounds?
Mm-hmm. Yes, so fluorine is actually, as it has having very extreme properties. So that's one of the reason why it's not very used in nature, and that's probably why it's very good in pharmaceuticals. The carbon fluoride bond is very strong, so it's very hard to break in the metabolism. So it works a lot as an inhibitor. So those extreme properties really changes the compounds which contain fluorine in its molecule. So everything is changed. The fluorine is very similar, for example, to hydrogen. So you can have a similar compound. However, maybe in some processes, this bond will not be broken. So that's why it is actually in many pharmaceuticals, and it's also why, as I have previously mentioned, why it's not very common in nature, is because fluorine actually is not very available in nature. So that's why it does not really appear. But in the chemistry, it was found out to be very useful, because the bond of carbon fluoride is so strong, and it comes with the other very important properties, which changes the properties of the pharmaceuticals.
Okay, thank you. And I guess now it's time for the last dreamer, Christopher Hendon, University of Oregon. Chris, the Zoom is yours, so share your dream with us, please.
What's going here? Okay. Thanks everyone for partaking today and for the invitation to speak about my dream. The title is A Chemical Fix for Bad Beverages. Well, I'm going to be speaking about drinks today. I actually didn't do my PhD in this discipline. I was Aaron Walsh's second PhD student at the University of Bath. And then shortly after I spent time there learning computational chemistry, I joined 3 groups at MIT. Mircea Dinka, who is a DREAM awardee winner, Yogi, who's a 2-time DREAMS finalist, and Yuri Rahman, who's a chemical engineer, where I spent 2 years learning a variety of different disciplines of chemistry and chemical engineering. And then shortly after that, I started an independent position at the University of Oregon, where I run a small research group with 4 graduate students, 4 undergraduates, and we study topics ranging from porous materials to solubility, to understanding pKa's of small molecules, all using computational chemistry. Now, we've been fortunate to be supported by the Office of Naval Research, the National Science Foundation, and then very recently the Coffee Science Foundation, which I'm going to talk a little bit about at the end. I can summarize our interest in the group sort of in 2 categories. On the left side, you can see these, we're interested in porous materials for energy storage and conversion. And the example I give here is a metal organic framework. So basically these are inorganic clusters. In this case, this is a nickel cluster supported by organic linkers. And then these metals can perform redox chemistry and other types of catalysis. So one example is this nickel center capable of oligomerizing ethylene, a small organic molecule that when it couples with itself will form butene, 1-butene, which is an important chemical feedstock in the polymer industry. Now, I could give obviously a whole talk on this, and hopefully I'll get to share some of that with you tomorrow. But I guess perhaps more related to what I'm going to talk about today is my other work in the coffee industry. And around 2014, I began an academic interest in coffee, and it ranges from topics at origin, so the production of the seed, all the way to the production of the brown beverage that you think of as coffee. And along the way, I've been awarded various things, including elected as a World Coffee Leader in 2016 and partaken in multiple Barista Championship teams. And so coffee really lays the foundation for one of my dreams today. But actually, this isn't the first time that I've been asked to dream. In fact, shortly after I started at the University of Oregon, I was invited to the University of Washington to partake in an Air Force Research Lab 2030 strategic meeting, where the Air Force was gauging interest from faculty members around the Pacific Northwest on various topics. And there, Major General Cooley asked a question. He said, to propose a technology, a weapon, or solution to use on our enemies that no one else would dream to propose. We went around the table of scientists, and some people had proposed using satellite-based weapons to kill terrorists before they even committed crimes, independent, of course, of the ethical problems associated with that. And then someone else, as we started to run out of good ideas, suggested that we had self-assembled metal endoskeletons so that soldiers could have retractable talons and have metal bones. And I felt like I'm pretty sure that I'd seen that Marvel movie. And then finally, we got to somebody who was undoubtedly a spy, who legitimately actually proposed that we should simply have more spies. But when Major General Cooley asked me what my solution was, I said, why don't we provide free renewable energy and gainful employment to all humans? At which point he put his hand on my shoulder and said, son, that shit is for the State Department. So I did what anybody would do, and I went to the local bar to drink my sorrows and feel disenchanted with the process. And it was here at this dive bar near the University of Washington where I conceived the second dream. Because as most American bars do, you'll run into a series of beer taps here on the left, several of which are extremely high quality beers. You'll also see that in this particular bar, there were 3 wine glasses hanging from the ceiling, which is either A, an indication that everyone drinks wine, hence all of the glasses are out with customers, or B, no one drinks wine and you definitely should not drink wine in this bar. And finally, you'll also note that there is a coffee urn sitting on the bar. Now, I indulged in each of these, but the beer was oxidized, the wine was overly acidic, and the coffee was too bitter. And then I conceived this dream. That I wish that there was my second dream was I wish there was a rapid chemical fix so that I could make all of these beverages that were thought to be high-end taste simply better. Now, beer, wine, and coffee are somewhat disparate in their relationship because you may drink one of these for breakfast, the other 2 you might drink later in the evening. Now, I'm not going to tell you which one that is, but the important point is that wine and beer both have been fermented. Beer and coffee both involve a roasted component. And so really these just canvas the spectrum of chemistry that one might indulge in while drinking these different beverages. But thinking more macroscopically about the beer, wine, and coffee industries, if you sum their GDP component, you get about 4.5% of the US GDP coming from these indulgences that we enjoy every single day. And importantly, in the beer industry, there are three primary brands that completely dominate the American market: Bud Light, Coors Light, and Miller Lite, which contribute 36% of the market share. And then in the wine industry, it may surprise you to learn that about 65% of wine is under $6. Now, I highlight coffee in blue here because there's not as centralized a market in coffee. It's a much more of a spider web of an industry, if you like. But also, coffee is dissimilar to beer and wine in the sense that you actually have to brew it at the end. But what I do want to point out on this slide is that a lot of people drink cheap beverages. But you would be misguided to think that this is a talk about those. In fact, it's absolutely not. What I'm actually going to talk to you about today is my dream of fixing bad flavors in products that had intended nuanced flavors, those that had character that were from a particular region in France, or a brewery, that, you know, a small brewery in the hills of Vermont. These are the ones that have terroir and perceivable craft. And so when we discuss flavor, we can break this down into three categories, where you have primary flavors, which are those like sweet, salty, bitter, sour, and umami. But more importantly, in the wine industry, sometimes people refer to, for example, grape as a primary flavor because it came from grapes. Then in the wine industry in particular, secondary flavor are those that are imparted by the process. And so this is primarily fermentation and oaking. But secondary flavor generation also exists in beer and in coffee. Of course, you can think of the roasting of coffee as generating flavors during the process. And then finally, you get to tertiary flavors, which are basically summarized as age-imparted flavors, or in other words, complexity. This simply takes time to generate these flavors. You can't do these from the process. But one thing that's often overlooked is that other chemical processes occurred over time as well. And so while you think of tertiary flavors as complex and interesting, equally challenging are the staling, failed containers that occur over time, and other taints that may not show initially in the production process. And so overall, one might simply summarize the problem is that if I pay more money for a product, I expect the product to be better, and hence I'm more disappointed when the product shows more variation. So let me frame my dream by way of example. Imagine on your wedding day, you were given a 1976 Penfolds Grange Hermitage Bin 95. This is a really big Australian Shiraz. And you go on to Wine Spectator and it tells you it's a drink now. And you read all the flavor notes and it sounds like something you're going to enjoy. But the last line causes you great concern, stating that bottles may be variable due to cork-related issues. Now we know the cork is there for multiple reasons. One, to keep the wine in the bottle, but two, to also keep the air away from the wine. So what could have happened? What could have given rise to this variation? So of course, one of the things that may have happened is exposure to air. So fresh wine has this deep red color. Well, fresh red wine, of course, has deep red color. And then over time, the pigments, and I give one example of a diol here, an aromatic diol, may oxidize, and you'll see that red color fade to sort of a more lustery orange. This process is thought to be catalyzed by solvated iron ions floating around in solution. And it goes via the so-called Fenton reaction, which is basically iron reacting with oxygen and shuttling electrons around, inevitably oxidizing oxidizable components of the wine and generating hydrogen peroxide sort of along the way, okay? So this isn't in itself bad. In fact, this I would call an interesting tertiary flavor or color. But another thing that could have happened from a failed cork is the wine could be corked. And what that would infer typically is the inclusion of a small compound, trichloroanisole. This compound, TCA, on the wine taster's flavor wheel is attributed to the flavor and smell, I should say, of wet dog and musty cardboard. And basically nobody likes this. But they say TCA, and then in parentheses, corked, because actually corked wine also achieves these flavors from other compounds like geosmin and 1-octene trione, okay? And so basically nobody enjoys these, okay? So let's say I have this bottle of wine and I open it and it's corked. The challenge then is how can I hide or destroy those flavors selectively in a complex mixture of water and other organic components and still achieve something tasty? And this is a very general but extremely challenging problem because, first of all, the compounds that are floating around in solutions that we actually drink may have arisen from various different processes. One could be the natural product, in other words, the quality of the grapes themselves or the quality of the coffee beans. Then there's the fermentation process that is common to all of the beer, wine, and coffee to some extent. And then there's the storage. And each one of these is going to generate different compounds and also have an opportunity to create taints. So on the right-hand side, I give one example. This is isohumulone, which is a compound found in fresh hops. But over time, the fresh hops will oxidize, and I show an intermediate of that here. This is the peroxide here. Okay, this obviously goes on to react and form other things. But the important point is that fresh hops taste vastly different to oxidized hops. Yet when we make a measurement of the hoppiness of beers, we simply take a UV vis measurement at a given wavelength. And both of these tend to sort of overlap in the absorption area in that region. So it's hard to make an assessment of quality based on a UV vis measurement. So to reiterate then, this is going to be a difficult problem to solve because we are going to try and target one compound that doesn't taste very good amidst thousands of other compounds solvated in water. And then within that, there are two challenges, is that many compounds are pretty similar in size, shape, and polarity, particularly in nature. And now, of course, they have different functionality. And to an organic chemist, you may think that they're really different because, you know, one of them is an alpha beta unsaturated alcohol, and the other one is an alpha beta unsaturated ketone, for example. But from a size perspective, they aren't that different. And so you get the idea that there are some chemical handles that we can play with, but there's some similarities between them as well. And then secondary to that is that water is, the water is an excellent solvent, so it's going to solvate these things. So it's not that easy to selectively remove them. And also water is reactive. So you can undergo oxidation and reduction chemistry of water. And so, of course, the general challenge here is going to be selectivity. Now, for corked wine, there has been a legend that exists that you could simply pour your corked wine over cling film, in other words, low-density polyethylene. And the concept is that you have low-density polyethylene and things that are nonpolar and would prefer to adsorb to low-density polyethylene rather than stay solvated in water will stick to it. And I give one example of a pyrazine here, but you can imagine doing this with trichloroanisole or any of the other taints that you might imagine. But of course, this doesn't achieve selectivity. I do say, though, that it works because according to wine experts who have performed this, at least they write about these in various articles on the internet, they say they can perceive a difference. If I ever come across a corked bottle of wine, I'm going to try this because it sounds somewhat interesting. I actually did have a go at trying to selectively remove target compounds using cyclodextrins. We had funding from the Australian Wine Research Institute back in 2014, where we proposed using alpha-cyclodextrin, which is just a cyclodextrin that contains 6 sugars in a ring. This one here in the middle is 7, and the one shown on the far right in this metal organic framework is 8. Now, it didn't work, but it may not have worked because the 6 cyclodextrin right here may not have actually had a big enough hole to put stuff in. And so you can see that either in our model wine, which is just ethanol, water, and then the taint, or in a non-fined white wine, we basically got no selectivity whatsoever, okay? Other people had found success using beta-cyclodextrins, and then I noticed that one of Fraser's postdocs, Fraser Stoddart's postdocs, and maybe he's still here, Fraser's still here, they reported actually a moth made of cyclodextrins, which is interesting because now not only are you targeting the inner area of the cyclodextrin itself, but also the porosity afforded by assembling this large structure. So maybe, who knows, maybe the CD moths that Fraser Stoddart reported have an application in selectively removing compounds from tainted wine, beer, and coffee. The other problem here is that positive flavors in one medium may be negative in another. And the best example is eugenol. So eugenol is a flavor that is imparted from oaking in wine, so we look for it because it tastes like wood. But if eugenol shows up in coffee, we disfavor it because woody notes in coffee are unpleasant. And then on the other hand, you might like 2-furfurylthiol because this compound is what smells of roasted coffee. But if you happen to have that in a wine, you might think there's some taint. And I give some other examples. So what I'm actually proposing is 3 general problems that need to be solved, independent of whether they were primary, secondary, or tertiary flavors, or whether it's in beer, wine, or coffee. I want a general toolbox to go after these problems. And I've distilled this down to 3 reactions: oxidation, reduction, and decarboxylation. And I give some examples electrochemically of what that would look like. So you can think of a reduced thiol as the free SH, and an oxidized version is forming that SS bond. And there are some other examples here. And in order to do so, I've just mentioned that we're thinking about electrochemistry, because it's a versatile toolbox that allows you to perform both reduction and oxidation in water, as well as more complicated chemical transformations where the electron is not the reagent, but actually nuclei move around, such as decarboxylation. And in a beautiful work by Phil Barron in a recent Accounts of Chemical Research paper, he did a better job than I could have of creating these figures. So thank you, Phil. Basically, an electrochemical cell is simply 2 conductive electrodes with some power source attached to it. And then in between these electrodes, you have positive and negative ions. And then when you apply a potential across these 2 electrodes, you can imagine pumping electrons into one electrode and depleting them from the other. And so the one that has electrons filling it will end up attracting positive charges, and the other one will attract the negative charges. But more importantly, you're able to then use those electrons and put them into the solution, react with species floating around in solution. So this is an example here where we're going to do an oxidation reaction where we're losing electrons from this amine to make this, this radical cation. So that's the oxidation. And on the other side, we're going to do a reduction reaction where we're going to take protons and turn them into hydrogen gas. Now, the nice thing about doing this in water is that you basically have an infinite source of protons, because water is, of course, H2O. So you're always going to be able to form hydrogen gas. Now, there is actually literature precedent for doing electrochemistry on beer, wine, and coffee. I show 2 examples. I've left coffee up for the time being. In one study in 2003, the authors demonstrated that fresh beer had a very different sort of redox features to older beer. So even after 2 weeks, you start to see certain features disappear. This is associated with oxidation, and then after 6 months, I mean, things are looking very different to that of fresh beer. Now, I don't actually know what the compounds are here, and that's obviously an area that we could improve upon. But the point is that electrochemistry can be used to quantify something about beer. And you can also do something with electrochemistry in wine. And so this is just a nice example showing that white wine and red wine, which is pretty low fidelity in my opinion, are different, okay? In the sense that you now have this feature showing up at around, I don't know, 650 millivolts versus silver chloride. And actually, you can even make the electrodes out of beer and coffee. And so in one example here, the authors presented first brewing the coffee when they got to the lab. They then took the coffee waste and then re-roasted it in the presence of zinc chloride in an argon atmosphere at 800 degrees Celsius and make N-doped porous carbon. This material is electrically conductive and is actually capable of performing the oxygen evolution reaction at about the same rate as that of bulk platinum. And so they go on to show that in a very nice paper in ACS Applied Materials Interfaces. In a different publication, a very similar process was performed, but for the lees of beer, so basically the spent yeast. And you can achieve N-doped carbon that way as well. But let's zoom out. Basically what I'm proposing here, and my dream is to be able to use electrochemistry and a simple potentiostat and a couple of electrodes to perform selective oxidation, reduction, and decarboxylation reactions to fix bad beverages. So these potentiostats are really cheap. Maybe one day we'll be able to use a mobile phone and have it in our hand. But the one on the left is from Thomas Maluk, who published in J-Chem Ed about a $100 device that will work perfectly fine to perform cyclic voltammetry or even single shot just electrolysis. And then going back to Phil Barron's work, a nice picture here shows, well, what do you want to do? Do you want to do oxidation or reduction? And this is where there's already a challenge, because you don't really know if you need to oxidize or reduce your bad wine, beer, or coffee. You need to taste it first and then teach yourself what you think is wrong with that beverage. But the important point is that at the anode, you're going to be doing oxidation, and at the cathode, you're going to be doing reduction, right? But let's say you wanted to perform reduction on a compound in solution. Phil Barron proposes using a sacrificial anode. This is problematic because if I were to perform that reaction, then I'm going to start filling my beer, wine, or coffee with zinc ions in the embodiment that I show here. Fortunately, we have a way of overcoming that. Now, for those that are more technically minded or have a bit of a nuanced knowledge of electrochemistry, I do want to make a couple of technical notes that the ionic composition of coffee, beer, and wine is non-zero. In other words, we don't need to add additional electrolyte to these solutions in order to pass current, which is good because that means we can just stick electrodes straight in there. Now, of course, these are much lower than perhaps what you do in the lab, but it'll still work. Secondly, the electrode materials that you choose to use are probably going to matter a lot. So you're going to have to play around with it, whether it's carbon, platinum, gold, nickel, or anything else. But the electrodes themselves have an electrochemical window, which is the window in which they're stable before the electrodes start reacting. But also, there's another electrochemical window associated with water. And the idea is that if you pump in too high of a voltage, you're going to start converting water into oxygen, which of course is an important chemical reaction. Well, going back the other way, is an important chemical reaction for fuel cells. And on the other hand, if you go at 0 volts by definition, you can start doing reduction chemistry and forming hydrogen gas, right? For this reason, this is why it's very difficult to then simply use electrochemistry to plate out sodium from water, because you're going to start doing this reaction before you get to sodium. And if you wanted to plate out, or if you wanted to bubble away chlorine gas, that's outside of this window as well, and so you won't be able to do that. And so you're limited to only doing redox transformations within this voltage range. Now, furthermore, there's going to be a tremendous amount of work associated with identifying the redox potentials for every compound that may exist in beer, wine, or coffee. But fortunately, we have a pretty good computational chemistry method for screening the redox potentials of various organic compounds. And there's no reason we couldn't use high throughput computing to get through all of the known compounds that have ever been reported and obtain a pretty good relationship between our computed redox potential, in this case, this is oxidation, and the experimental oxidation potential. And if you have confidence in this, then we'll be able to start to understand how we can achieve selectivity by hitting different ranges. So I want to give you a couple of examples. Imagine you have a liquid, for example, an Ethiopian coffee that's been naturally processed and therefore has unusual fermented fruity flavors. And you want to perform electrochemistry to intensify the secondary flavor of blueberry that was imparted from the natural process. And here on the coffee taster's flavor wheel, you can see that blueberry is sitting under the berries region, which is a positive region, an area of flavors that people like in coffee. Now, maybe you've never experienced it, or maybe you don't like it. And so the point is, this is still inherently general. It's not inferring that you must do this. But the idea is that you might find that in a fresh coffee processed in Ethiopia, it'll be very high in 2-hexene-1-all, which does taste like blueberry. But actually, 2-hexanal, which is the oxidized version of that, tastes even more intense of blueberry. And of course, this would only occur over time because this is an oxidation process. But there's no reason you couldn't do this electrochemically by simply submerging 2 electrodes and then performing the oxidation to target the transformation of the alcohol to the aldehyde while simultaneously reducing protons into hydrogen gas. Of course, the hydrogen gas will bubble away. I mean, who cares about that? The important point is now you've got a really intense blueberry flavor, and the potential that you'll do that at is relatively facile, so definitely achievable in water. What about an electrochemical reduction? This is a little bit more complicated because, as we mentioned before, typically the oxidation process would involve the sacrificial anode, in other words, stripping this electrode into the solution, but that's not ideal. Well, one example is undoing a tertiary aging. When fresh wine is processed, you arrive at methyl anthranilate, which is this compound here, in this case intensely of grape. But over time, this oligomerizes and forms compounds that look like this. And this is the trimer, but there's also the dimer and other higher order polymers of this thing. Now, this large compound doesn't taste like grape at all. In fact, we don't have a flavor note. We can't perceive it, but the smaller one does. Now, maybe you like that. Maybe the grape needed to de-intensify a little bit by forming this trimer. But perhaps maybe the wine has fallen off a cliff and now no longer tastes of anything. And you might want to undo that process a little bit. And so what you could do is then do reduction. You could back reduce this or undo the tertiary process, getting back to our methyl anthranilate, which has an intense grape flavor, and doing this selectively so that you don't take away all of this and don't, create too intense a grape. But at the same time, you're going to have to then turn water into hydrogen peroxide. But you might remember then that the hydrogen peroxide actually partakes in the Fenton reaction that I mentioned at the beginning, which is important for aging wine. So actually, this hydrogen peroxide here, it may actually have an added benefit of sort of creating a different hue or something additional. And I don't know. Remember, this is the dream competition. So my dream is to be able to control all of these things. Now, finally, the electrochemical decarboxylation is an important reaction because it will, in principle, reduce tartness, sourness, and overly acidic beer. And there's been this recent trend in drinking these terribly sour beers, particularly those of the Lambic style from Belgium. And I'm sure that some people like them, but we've all had the experience where it makes your face want to melt off. And so basically, an electrocatalytic decarboxylation is a way of lowering the content of organic carboxylic acids through decarboxylation, which is thought to proceed first by a hydrogen atom extraction, like we show here, giving rise to a carbon-centered radical. Now, depending on the concentration, if you've got a lot of those carbon-centered radicals around, they may couple and you might end up doing a Kolbe electrolysis reaction where you form some new compounds. Who knows? They may taste good. We're going to have to try that out. But at dilute concentrations, one could imagine simply bubbling off CO2 and then having a H dot add into this thing, forming the new alkane, or at least the hydrocarbon terminated compound. And so let me give you an example of the real problem, is that for these Belgian-style lambics, they may be exposed to both Brett and lactobacillus, which are two different enzymes, or bacteria, that will chew up the various compounds in the beer brewing process and spit out little things like ethyl lactate as well as lactic acid. Now, it's a misconception that Brett imparts sourness. Brett does impart flavor in the form of this ester, but actually the real sourness in these beers come from typically uncontrolled lactobacillus. And so if you have too much of the lactic acid, you may want to electrochemically decarboxylate it. And so here, there are two options. Given that hydrogen atom extraction must go first, you can either imagine targeting the alcohol or the carboxylic acid. Well, if you target the alcohol, who knows what's going to happen? You're going to get a variety of products. But I think that the carboxylic is going to be more stable because you can delocalize that radical. And what's neat about this is that if you do decarboxylate this product, you end up with ethanol and CO2. And this beer was already fizzy, and you made some more alcohol, which sounds great. But if this doesn't work, right, I do have a backup plan, which is you could simply just titrate with buffer, like bicarbonate, for example, and turn off some of that acidity. But the problem with that is we don't know what the conjugate base of this acid tastes like. And so we just simply know that on the beer taster's wheel, under the section of bad and infection and wild bacteria, which are all sort of like unpleasant, we have sour, funky, acidic, and lactic. And these are sort of the areas, the bad panels, if you like. And so I really don't know what's going to happen if we do this titration. But I want to take your mind back to that original bottle that you received on your wedding day, the 1976 Penfolds Grange Hermitage Bin 95. And let's assume that it has been tainted by 1 octene 3-ohm. In other words, it's cork tainted by this particular compound. Well, this compound smells and tastes of wet dog and cardboard. But you know that with a 2-proton, 2-electron reduction, you achieve 3-octanone, which is, in principle, it's nectarine and lavender. And so what I'm really saying is you're going to take this panel, which is bad, and convert it over to this panel on the wine tasting wheel to nectarine, which is good. And how might you do that? Remember, you just need a potentiostat and a couple of electrodes. And so here's this guy opening that bottle of wine, and he's thinking about what electrodes he should use for this bottle of wine he got on his wedding day. And he needn't look very far because he's already holding the electrode that I propose. Simply take off your ring, as long as it's one of these precious metals, and maybe use a pencil or some other material, and you should, in principle, be able to perform this reaction. But what about the beer, wine, and coffee that was not overly complicated at the beginning? These market-dominating brands, Bud Light, Coors Light, Miller Lite, and the cheap wines. Well, they don't really have that many problems because they don't have that much complexity. So you basically know what you're getting when you order these sorts of things. But what if you did want to impart flavor? Well, you could dissolve something that doesn't have flavor and then turn it on by doing that electrochemical reduction. And I give that one example again of methylanthranilate simply by way of example. And there's plenty of other opportunity here to impart different flavors into these starting points. So very briefly, I want to share with you my progress towards the dream. Is that recently I was funded by the Coffee Science Foundation. And the idea was to improve the so-called brew control chart, which is simply a relationship between the mass of coffee that you extract per gram you use to brew it and the concentration in which that extraction exists floating around in solution. And somewhere in there between 18 and 22% extraction yield and 1.2 and 1.4%, it's thought that coffee, at least a cup of coffee, tastes ideal. But I dispute this because how can that be if we're just measuring mass when we know from the 1901, the very first Nobel Prize, that temperature affects the equilibrium constants of given reactions? And importantly, I show here my experiments with caffeine brewing coffee at different temperatures. Whereas at high temperature, I'm able to extract about 15 milligrams of caffeine per gram of coffee I used. But at 20 degrees Celsius, I can only extract about 9 milligrams of caffeine per gram of coffee that I used. Yet, at all temperatures, I'm able to extract within the margin of my measurement all of the caffeic acid. So the relative ratios of these are changing. And that means I'm not going to be able to make an assignment of the flavor using simply extracted mass. And so what I proposed and what we're working on is the development of homemade electrochemical setups that we distribute to the coffee industry in order to make these measurements of maybe total oxidizable or reducible content to be able to assign qualities in coffee, not only quantities. And I want to note that we intend to have these electrochemical setups in the hands of about 50,000 individuals and cafes around the world by 2023. And I intend to do that by leveraging my role in the coffee industry and having all these wonderful people who are prepared to experiment and learn together. So let me summarize that my dream is to have a chemical fix for bad beverages. What was initially inspired by Major General Cooley's disdain with my proposal for energy sparked my interest in solving the problem in the dive bar. And there are several opportunities. Of course, we're going to have a generalizable approach to perform reactions in water, and inevitably you might enjoy these beverages. But there's going to be challenges because, of course, selectivity is always going to be an issue when you've got thousands of different compounds. And of course, also, the rate may be slow. So maybe you could make this correction, but if it's going to take 2 days to do it, it's not going to be overly enjoyable. The beer will be warm, the coffee will be cold, and so forth. And I've also offered you a way to improve those beverages that may not necessarily have been excellent at the very beginning, the commodity grade beverages that we also enjoy. But finally, in this process of thinking about the dream, I realized an additional dream, which was if I can get these electrochemical setups out to the general public, then what we've really done is we've achieved an improvement in scientific literacy across the board while involving and engaging people in science at the frontier, at the level of our dreams. So with that, I want to thank a few people. There's been several people in the industry, baristas, beer brewers, winemakers that have helped me over the years, coffee roasters, and academics. But importantly, I want to congratulate my fellow Dream 2020 finalists. I learned a lot, and it was very thought-provoking throughout the day. Also, thank you for organizing this wonderful event, because this year has been difficult for us for a variety of reasons, and it's been really nice to look forward to thinking about something fun and enjoyable at the end of this year. And then finally, the two people who really helped me develop this talk, Alex Murray, a professor at Kent, who really fleshed out some of the electrochemical challenges, and then Carl Brozek at Oregon, who is basically an excellent professor here at UO, but also one of my best drinking buddies. So thank you very much. I'll happily take any questions.
Okay, thank you, Chris. Next time I drink coffee, wine, or beer, I will keep thinking about you. The first question is from Pavel Jungwirst. Pavel, please.
Put it on a sort of a lighter tone. I will accept the challenge, and I'll play the devil's advocate, but take it as, we're on the same tone. I happen to be an amateur wine grower and winemaker myself for a few years now. And there are two things you learn when you're making wine. So I'll focus on wine. I know much less about beer and coffee, although I'm Czech, so I should know about beer.
Sure.
So the first thing you learn about wine is it's 90% is the quality of the grapes and 10% is the process. And the second thing you learn about this remaining 10% is that you can screw up a lot, but you can't fix too much. Okay, so that's where I see a major challenge here, right? And another way of maybe directing you into some thinking or answer will be, I've noticed there were attempts to make synthetic wine. So just make wine without any of the grapes, right? Just mix the chemicals. And the results were, I mean, unimpressive so far, right?
Questionable.
I'm throwing this on you, and I just want to see how you handle this.
Sure. So interestingly, I agree with you entirely, not only in wine that the quality of the natural product, the grapes, is the primary determinant for the quality downstream. That's actually true in coffee as well. It's a widely established, I think probably in beer also. And indeed, you know, there's not really a way that you're going to take a bad quality grape and turn it into a beautiful wine. And I was really trying to avoid motivating this talk by taking low quality products and upgrading them, but rather taking the higher quality products and rescuing them in case something terrible had happened. But you're also right that you can basically ruin anything good along the way. And that's really where determining what you did, whether somehow they oxidized or somehow bacteria was introduced that you weren't expecting, that's really difficult. And I'm not sure that we have an analytical method to assess where the problem occurred after you've produced the beverage. A great example of that is you think about a smoke-tainted grape, in the sense that you don't really know when you produce the wine whether it's going to exhibit these characteristic smoky flavors until much later down the road, a couple of years, and then it shows smoke and you think, oh, this is truly terrible. But was it smoky because of a fire, or was it just that's what that grape was producing in the first place? We don't really know. So that was part one.
And part two was the synthetic wine. So just like, what is your thought about the synthetic wine efforts?
Yeah, so it's not only in wine that they made synthetic wine. There's a company right now that's trying to make synthetic coffee because of crop sustainability issues. And I've had I don't know if luxury is the right word. I've had the opportunity to try the synthetic coffee, and I can tell you much the same as your experience with the synthetic wine, that there is room for improvement. But that doesn't mean that it's not a good product, right? It just means it's a different product. And I really hope that it didn't come across like I'm trying to standardize flavor or trying to even tell you what does or does not taste good, but simply giving you a general toolbox to be able to modify the chemistry in these complex liquids. And indeed, I think you actually summarized my dream, right? The dream is this is really difficult. But that was the challenge.
Thank you. Awesome. Thanks.
Okay. The next question is from Daniel Gryko. Daniel, please.
Chris, you are the dreamer. I mean, to dream that people will come with a little potential state to the restaurant. I mean, this is really, really broad dream, and I admire you for that. But I would like to ask you the chemical question, because you mentioned these challenges, selectivity, and even though, let's say in wine, the total weight of the complex organic molecules is relatively low, but the number of them is overwhelming.
Right.
And you start to oxidize, you start to reduce. There are books about oxidation and reduction. So is it, I mean, as a dream is great, but is it at least a little bit realistic to be able to fix the smell or fix the taste with such a complexity inside, which is intrinsic, because you cannot remove this complexity, right? But it's like it's a mixture.
Yeah, that's right. So you basically summarized the challenge. I mean, you've also asked a technical question, so I'll give you a slightly technical answer. That one of the challenges that was overlooked in my talk is that if we don't have a reference electrode, we also don't know what potential we're operating at. So in principle, we could be just hitting it with huge voltages and fully oxidizing or fully reducing everything in the solution, and everything will, who knows? I mean, I don't want to taste a fully oxidized wine, right? So selectivity is always going to be very difficult. even if we knew all of the potentials, this would still be very difficult. But I've also seen very difficult chemical transformations by groups like Phil Barron's group out of La Jolla, perform difficult and selective transformations even within a molecule that has multiple different redox active components. So if it can be done within a molecule, it could in principle be done within two different molecules that have different redox potential. But being able to do it within 1,000 compounds, it's a dream. I don't know. I just don't. I don't know how much work.
Yes, undoubtedly, one thing is clear that synthetic electrochemistry is rising, and Phil Barron is sort of prophet of it. We have just started it in our institute, essentially last week. So that would align with a trends. Yeah. All right. Good luck. Good luck.
Yeah. Thanks.
Okay, so Renata Biles, please.
Processes, and with your glass of wine, there will be oxygen, a lot of oxygen, and the thing that you will reduce will be oxygen instead of your compounds that you want to suitably reduce. How do you want to eliminate oxygen?
So
in the wine place.
Yeah. So I've done a very small amount of thinking about this problem. With some of the wine professionals that I've worked with, they're making assessments of the contents of the bottle without actually removing the cork by sort of putting in a cannula and then back flushing with nitrogen. So you're not reintroducing oxygen to the bottle. Okay. But why that's important is that there's iron already in the wine itself, and there's a little bit of oxygen in the headspace between the cork and the liquid. And over time, that gets rapidly chewed up, and so there's a bit of oxidation. But there's very little residual oxygen in the bottle of wine before you pull the cork. So in principle, you could do this with wine if you had a way of pulling out a sample or doing the electrochemistry directly inside the bottle before you remove the cork. But that would be very difficult to, you can see there's some sort of fundamental challenges, but at least we know there wouldn't be too much oxygen, not too much within there. And I can point you to work from Andrew Waterhouse at UC Davis, who has done some beautiful studies on the sort of the decline in oxygen content in the headspace over a very short time.
Yeah, I was just speaking about the glass of wine.
Yeah, so when you put it in the glass, yeah, I don't know. I mean, you got fundamental challenges. Yeah.
Okay.
So I have.
Okay, the next question is from Bogusław Buszewski. Bogusław, please.
As you are talking about selectivity, and another very important problem is specificity, because we have bouquet, so some volatile compounds, and the interference of some compounds strongly influences the processes which you want to control. And you said headspace. This is, for example, volatile compounds analysis, but which one will be more important for the realization of this reaction? This is what first question. And another one, this is connected with speciation, because you present also some different metals, for example, which participating in this process. So what about that?
Yeah, so am I understanding your first question correctly in the sense that some of the compounds that we'll be looking at are indeed volatile, and that will pose fundamental issues with this analysis. Yeah, so one of the difficulties with these volatile compounds, particularly this is really important for coffee aging, is that once you brew the coffee and then the aromas have left the cup, they're not there anymore. It's not like you can simply recover them. And so this, the chemical process we're discussing here in the headspace I was referring to in my previous answer.
But not only, Christopher, I'm so sorry. So this is not only for the coffee, but also the bouquet of wine play important role.
Sure. Yeah. So the reason I mentioned coffee is simply because of the elevated temperature. So it just,
I prefer wine, red wine.
Well, yeah. Okay. So for example, for wine as well, right? You're going to lose volatiles.
Yeah. Okay. No.
Right. And there's not really a solution. If the chemistry is in the atmosphere now, I don't have a solution. I don't have a way of, at least not within the remit of what I'm talking about.
Yeah, but for this process, the metal participating in such is very play important role. This is bottleneck for some process, electrochemical processes.
Right. So, I mean, you're right. I don't really have a solution to the problem for those that are forming extremely volatile compounds. There's basically, it's one of the issues that we face in even fundamental electrochemistry.
Yeah, but this is the question of the artificial electrochemistry, so like Smurf generator or I don't know what.
Right. Okay. Right. So yeah. So I don't have a good answer for that question simply because I'm not sure that there is a good answer to the question.
No, this is interesting for us from the point of view of the breath analysis, for example, medical diagnosis.
Right.
The composition of this.
Right.
Thank you. It was connected with speciation of the metal, for example, the influence of this. But
yeah.
It was connected with the first one.
Yeah. So it's the same response. We just do some work.
Iron, for example, particularly iron.
Yeah. So I actually don't know what is thought to be the speciation for iron, but
but also copper, So this, yeah. Thank you.
Okay, so we are coming to an end of today. Yunyan, Claudia, Chris, Pavel, Ivana, thank you very much. And I don't know whether you've noticed that you are all from different countries of origin and you represent different countries. So Yunyan is from China, but he represents Northwestern University. Chris is from Australia, representing the University of Oregon. Claudia is from Italy and representing University of Cambridge. Pavel is from Poland and representing France University de Strasbourg. And Ivana is from Slovakia and representing Vrije University in Amsterdam. I really love it in science that from the very start of the modern science, we could communicate very easily over the borders. Newton exchanged the letters with Leibniz and they, of course, were writing in Latin as we communicate now in English. And so dreams also don't see the borders. Now you are all winners because tomorrow you will get the top 5 awards. So let's congratulate you, right, guys? But who will get the statue? I'm not a good guesser. We have a committee of 18 very strong personality professors. And so usually when we start the discussion, each person that took part in the finals got the votes. And then there is a really long discussion where you tell not only for whom you vote, but also why you vote. And then we make like 5, 10, 17 rounds. And finally, together, we agree on a single person. So guys, it's always kind of a lottery. I will keep my fingers crossed for all of you. So we will have a meeting of a committee at 10 o'clock a.m. and we'll have 8 hours to decide. It's like getting a new pope or something. See you tomorrow, same time, 5:45, and then the director of the institute will hand you all the rewards and they will also hand you the statue. And then we'll hear one of the lectures, but all the other 4 are already invited to our institute to give the lectures that others cannot hear tomorrow, okay? And, what can I say? 18 strong personalities will choose one person out of you 5, guys. But for me, you are all winners. Thank you very much. It was a wonderful evening, so thank you. Cheers, bye.